everyone, and welcome to this entire A-level video for the year 13 topics, which are topics 12 to 19, which will cover paper 4. In this video, I go through everything you need to know in terms of the theory for that A-level exam. And if you do want even more help preparing for that exam, then don't forget to check out my A-level notes, which covers all of the theory for the entire A-level, topics 1 to 19, key marking points, key terms, and also examiner tips as well. So that is linked below for you. But for now, let's get into the video. So topic 12, let's get into it. And it's covering energy and respiration. So first of all organisms require energy, and that is for metabolic processes such as active transport, where you're moving substances across a membrane, or other types of movement such as using a cilia or flagella or muscle contractions, and also for building polymers, so protein synthesis where amino acids are then converted and joined together to make proteins. That requires energy, as well as DNA replication, joining together the DNA nucleotides. And energy can't be created and it also can't be destroyed, and instead organisms use energy that is coming from bonds. And the opposite of that is then making bonds to create molecules like polymers. That requires energy, but breaking them releases energy. So photosynthesis is an example, and this converts energy from the sun into actually creating bonds, whereas respiration, this involves breaking bonds such as glucose, which is the respiratory substrate used to ultimately create ATP. And that ATP can then be hydrolyzed into ADP and PI, and when that bond is broken between the inorganic phosphates, that is what releases the small manageable amounts of energy which can be used in metabolic processes. So let's take a closer look at the structure of ATP. Now it's known as a nucleotide derivative because it's got very similar components to a nucleotide. It has this pentose sugar, it has a nitrogenous base. The key difference is instead of one inorganic phosphate group, it has 3 inorganic phosphate groups. And you need to know then that it is an immediate energy source for biological processes. Metabolic reactions in cells have to have a constant supply of this ATP, so they have the energy for reactions. And it's the bonds between these 3 inorganic phosphates, which when they are broken, the energy is released, and that's used for chemical reactions within the cells. So this structure then, we briefly touched on it because we said it contains a nitrogenous base. In ATP, that nitrogenous base is always adenine. And nitrogenous means it contains nitrogen, which we can see here inside of those rings. The pentose sugar is always ribose, and then it has 3 inorganic phosphates. So we can see here those bases, or that base adenine, the ribose, and the 3 phosphate groups. Sometimes you might see it called adenosine triphosphate. Adenosine is the term for the adenine and ribose together. So it's adenosine with 3 phosphates. That's why it's adenosine triphosphate, or ATP. And the level of detail that you need to know it in is actually here. You don't need to know all of the atoms within it. You just need to know that you have the nitrogenous base bonded to the pentose sugar, which is ribose, bonded to these 3 inorganic phosphates. And the phosphate groups are described as being inorganic because they don't contain any carbon atoms. And for this reason, in chemical reactions, the symbol used to represent the inorganic phosphate is Pi, meaning inorganic phosphate. So a bit more about ATP then. And this time we've looked at the structure. Now we're going to look at the creation and hydrolysis of it. So ATP is made during respiration from ADP, which is adenosine diphosphate, by the addition of an extra inorganic phosphate. And that's a condensation reaction using the enzyme ATP synthase. And that's what we can see here, ADP plus PI, that results in the production of ATP plus water. And it's a condensation reaction, so that's why we've got plus water. Water's been removed. This is a reversible reaction. ATP can be hydrolyzed back into ADP plus PI. It's a hydrolysis reaction, so water is added, and with the help of the enzyme ATP hydrolase catalyzing this reaction, it will then split or hydrolyze ATP into ADP plus PI, and that's when energy is released, when the bond between those inorganic phosphates is broken. So this is done immediately. You don't actually store ATP. So that's why ATP is known as an immediate energy source. You also get this energy very rapidly because you only have to hydrolyze or break one bond to release that energy. And as well as releasing energy to the surroundings, ATP can actually transfer energy to other compounds. Because if you add that phosphate onto a different compound, that compound will then gain the energy. And that's what phosphorylation is. It's when you add an inorganic phosphate group onto a molecule so it becomes more reactive or it gains energy. So technically, this is actually a phosphorylation reaction because ADP is gaining a phosphate group to become more reactive or to have more energy in the form of ATP. And we'll also see this happening in the first stage of aerobic respiration because glucose gets phosphorylated as well. So energy values of respiratory substrates. Near the start of this video, I said that glucose is a respiratory substrate, but you can actually use other carbohydrates and lipids and proteins if there aren't other carbohydrates available. And all of these can provide energy to be used in respiration, but they all have slightly different energy values per gram, and that's due to the variations in the chemical structures and metabolic pathways. And that's what I've got summarized here on this table. So you might want to pause at this point and just copy this information. So we've got carbohydrates, lipids, proteins, glycogen, or if it's in a plant, it'd be starch. Glycogen's only for animals, and then alcohol. So we've got the energy yield per gram. So we can see you get the most energy from lipids, then alcohol, and then carbohydrates, proteins. Glycogen and starch are actually carbohydrates. This is referring more to sugars. And you can see the metabolic pathways and then the conditions of use. So that's a good one just to pause, copy down, or turn into flashcards if you want. So the respiratory quotient then, or RQ, of each respiratory substrate can be measured using a respirometer, which is one of the practicals that you need to be familiar with. And you divide the volume of carbon dioxide produced by the volume of oxygen used. So that would be your formula, the carbon dioxide produced divided by oxygen consumed. And then you can use that to compare different substrates as well. So now we're going to move on to 12.2, which is respiration, both aerobic and anaerobic respiration. Starting with aerobic respiration, there are 4 key stages: glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation. You need to know where all 4 of those stages happen within the cell. Glycolysis happens in the cytoplasm, but the other 3 stages all happen inside of the mitochondria. The link reaction and the Krebs cycle are within the mitochondrial matrix, which is shown in blue on this diagram, and it's the fluid-filled center. Oxidative phosphorylation, the final stage, happens on the cristae, which is the name for this folded inner mitochondrial membrane shown in yellow. So let's start with glycolysis and the first stage. And this stage is where you are producing a small amount of ATP, but you also use a small amount of ATP. We said already it happens in the cytoplasm, and this stage doesn't require any oxygen, and therefore it occurs in both anaerobic and aerobic respiration. And there are 3 key steps that you need to know. The first step is substrate level phosphorylation, and this is when the glucose, which is the respiratory substrate, has 2 phosphate groups added to it from 2 ATP molecules. And that makes it a far more reactive molecule. And that's then what happens in the next stage, that phosphorylation. Once you've added those 2 phosphate groups to glucose, it creates fructose, 1,6-bisphosphate, which is a 6-carbon compound still, which is like glucose, 6-carbon compound. But because it's gained that energy from the joining or the bonding of those 2 phosphate groups, it makes it unstable, and therefore it splits immediately into 2 3-carbon compounds known as triose phosphate, or TP. Lastly then, those TP molecules, both of them, are oxidized, and they're oxidized by removing a hydrogen. And the reason removing a hydrogen is oxidation is a hydrogen atom is 1 proton and 1 electron. So oxidation is the loss of electrons. If you remove a hydrogen atom, you are removing an electron. So it's oxidized by the removal of a hydrogen atom, and that will then form 2 pyruvate molecules. Now also in this oxidation, we're going to create 2 reduced NAD. NAD is a coenzyme, which is a molecule that can pick up and transfer hydrogen from one part of the reaction to another in respiration. And we have NAD gaining the hydrogen that's been lost, and therefore it's gained an electron. And reduction is gain. So if you think of oil rig, oxidation is loss, reduction is gain. So reduction is gaining an electron, and if you pick up the hydrogen, you are gaining an electron. So you can call it NADH or reduced NAD. And that stage 3, also releases 4 ATP molecules. But that means we have a net gain of 2 ATP from glycolysis, because we had to use 2 at the start. Take that away from the 4 that are made, we have a net gain of 2. So that's the stages written down. Let's have a look at it as a visual representation so it makes hopefully a bit more sense. So we start off with glucose, the 6-carbon compound. We then have that substrate level phosphorylation stage where we said we're going to be using 2 ATP molecules to add on 2 phosphate groups. So we phosphorylated it to form fructose 1,6 bisphosphate. That is now very reactive and unstable, so it splits straight away into 2 molecules of triose phosphate. That triose phosphate is then oxidized into 2 molecules of pyruvate, and it's oxidized by removing hydrogen, which is picked up by NAD to form reduced NAD or NADH, and in doing so, 2 ATP molecules are created. That happens on both of the triose phosphate molecules. So overall, the products we get from glycolysis are 2 3-carbon compounds of pyruvate, a net gain of 2 ATP, so although we made 4, we had to use 2, and 2 reduced NAD, or 2 NADH. And the pyruvate and the reduced NAD are going to be used in the later stages of aerobic respiration. So here we have our glycolysis happened in the cytoplasm. The next stages happen inside of the mitochondria. And as we just said, the pyruvate and the reduced NAD are going to be used in later stages. So those now need to be transported into the mitochondria, and they get transported in by active transport into the mitochondrial matrix. The next stage then is the link reaction, and this is when the pyruvate from glycolysis is oxidized to acetate. We have another NAD, which is going to pick up the hydrogen from this oxidation to create reduced NAD. And that's what we can see here. The pyruvate is oxidized into acetate, and we know it's oxidation because we have made a reduced NAD or NADH, which means the hydrogen must have been lost from pyruvate. You actually also have decarboxylation, which is the removal of a carbon in the form of carbon dioxide. So that's why we go from a 3-carbon compound pyruvate to a 2-carbon compound acetate. Now, acetate can't enter the next stage, which is the Krebs cycle, without the help of coenzyme A. So acetate then combines with coenzyme A to make acetyl-CoA or acetyl coenzyme A. And that is the link reaction. We don't have any ATP made in this stage, but we do have acetyl-CoA, carbon dioxide, and reduced NAD. And I've written 2 times for all of these because although we only get 1 NADH, 1 carbon dioxide, and 1 acetyl-CoA from the link reaction happens twice for every glucose molecule because we had 2 pyruvate molecules made in glycolysis. So the link reaction will happen twice for every glucose molecule. So that's why we've said 2 times those products. Next then is the Krebs cycle, which is still happening in the same location, which is the mitochondrial matrix. So here we can see the link reaction, and that's going to lead us into the Krebs cycle. And the first step of the Krebs cycle is oxaloacetate, which is a 4-carbon molecule, is going to combine with the acetyl-CoA. And because we've got a 4-carbon reacting with a 2-carbon, we get a 6-carbon molecule, which is citrate. And you do need to know the names of those compounds, oxaloacetate and citrate. At this point, the coenzyme A has served its purpose of helping the acetate react with the oxaloacetate, and therefore it's released and it goes to be reused again in the link reaction. We then have a series of redox reactions. There's actually lots of intermediates here, but you don't need to know any of those intermediates. You just need to know that there's a series of redox reactions, And that is because the carbon compounds are going to be oxidized, and these coenzymes are therefore going to be reduced. They're picking up the hydrogen that's being released from the carbon compound. So we end up making lots of reduced coenzymes. We have 2 carbon dioxides that are going to be lost, and that's how we go from a 6 carbon back to a 4 carbon molecule. And then we also have 1 molecule of ATP being produced at this stage. So from one round of the Krebs cycle, we get 3 reduced NAD, 1 reduced FAD, which is another coenzyme that picks up hydrogens, 1 ATP and 2 carbon dioxides. But much like with the link reaction, the Krebs cycle happens twice for every glucose molecule because we have 2 pyruvate made, therefore 2 acetyl-CoA, therefore the cycle happens twice. So for every glucose molecule, we have 6 reduced NAD, 2 reduced FAD, 2 ATP, and 4 carbon dioxide molecules. So this is just splitting it up into those 2 stages, the link reaction and the Krebs cycle. So summary so far, we've gone through glycolysis, which happened in the cytoplasm. The pyruvate and the reduced NAD were actively transported into the mitochondrial matrix, which is where the link reaction and the Krebs cycle happened. So far, if we total up what we have produced in terms of coenzymes, because those are what are vital for this final stage, oxidative phosphorylation, we have made in total 10 reduced NAD from 1 glucose molecule and 2 reduced FAD from 1 glucose molecule. And those reduced coenzymes are really important for this final stage, which is oxidative phosphorylation. And this is where most ATP is created. So it involves an electron transfer chain, involves the movement of protons across the inner mitochondrial membrane or the cristae, and we get ATP being produced by ATP synthase. So let's go through what's happening then. In the mitochondrial matrix, that's where we've just had the link reaction and the Krebs cycle happening, producing lots of those reduced coenzymes. Those reduced coenzymes are now going to have the hydrogen removed, which we can see here, the hydrogen's been removed, and that hydrogen splits into the protons and electrons. And in this diagram, the electrons are represented by these star-shaped suns, and the protons are shown here. So the electrons are picked up by proteins embedded within the inner mitochondrial membrane. And this is the electron transfer chain, because the electrons then transfer along these proteins. As those electrons move from protein to protein, they release energy. And that energy is used to pump the protons from the matrix through these proteins into the intermembrane space. And therefore we end up with lots of those protons, which are hydrogen ions, all on one side of the membrane. And it's in the intermembrane space, which is the gap or the space between the inner membrane and the outer membrane. So we get this electrochemical gradient, which means we've got a lot of a charged molecule. So that's what's electrochemical gradient on one side of the membrane. And that then results in the protons, these hydrogen ions, moving back across the membrane by facilitated diffusion down their concentration gradient. And the only protein they can move through due to complementary shapes fitting together is ATP synthase. And as those protons move down their concentration gradient back into the mitochondrial matrix through ATP synthase, that then causes the catalyzation of ADP plus PI into ATP. So as all of these protons move back down their electrochemical gradient, that's how we get lots of ATP being produced. Now, the final thing to be aware of is the role of oxygen here. And oxygen is known as the final electron acceptor. And that's because as these electrons move along the electron transfer chain or electron transport chain, at the end of the chain, if oxygen wasn't here to pick up those electrons, then they would stay in that protein, and therefore no more electrons can move along the chain. And if you don't have electrons moving along the chain, you don't have energy being released to pump the protons, you don't have an electrochemical gradient, and therefore you don't get ATP being produced. So it's really important that oxygen is this final electron acceptor, meaning it picks up those electrons so that the electron transfer chain can continue and we still get ATP being produced. And that oxygen picks up the electrons. It also picks up some of the protons, and that is how water is produced in aerobic respiration. So we then move on to anaerobic respiration, which is respiration in the absence of oxygen, and it only occurs in the cytoplasm of the cell. It involves only glycolysis from the stages we just saw, and the pyruvate that's made in glycolysis, in anaerobic respiration, it's reduced instead of oxidized. And in plants and microbes, it's reduced to form ethanol and carbon dioxide. In animals, it's reduced to form lactate or lactic acid. And this is done by the pyruvate gaining the hydrogen from the reduced NAD that were made in glycolysis. And the reason that needs to happen is that it's going to oxidize the reduced NAD back into NAD, and that means it can then be reused in glycolysis, so you can still at least have glycolysis occurring to produce 2 ATP molecules. So let's have a look at that represented visually. We said glycolysis occurs, so that's what we've already just gone through. And then this time the pyruvate is reduced. And we can tell it's reduction because that NADH becomes NAD, which is the oxidized version. And that hydrogen is picked up by, and that would be 2 NADH here, because it's going to be picked up by 1 each by these pyruvate molecules. And that is then going to reduce it into lactate or lactic acid. And that is actually toxic. Lactic acid is what causes muscle fatigue, but it is worth making that toxic molecule so that the NAD is regenerated and glycolysis can still occur to make at least some ATP. And anaerobic respiration can't happen indefinitely because that lactic acid would denature enzymes involved in this stage, and therefore it would then stop. And that's where we get this oxygen debt, where you need to take in lots and lots of deep breaths to get enough oxygen to fuse it into the bloodstream. And that oxygen can then break down the lactic acid in the liver back into non-harmful substances that can be used as respiratory substrates. Similar in plants and microbes, but instead of making lactic acid or lactate, we have ethanol and carbon dioxide being produced. So in terms of the efficiency of these reactions, one reduced NAD can result in a yield of 3 ATP molecules in that oxidative phosphorylation that we just saw, whereas one reduced FAD can result in a yield of 2 ATP molecules. And if we think back to the fact that we said from one glucose molecule, we create 10 reduced NAD and 2 reduced FAD, that results in 34 ATP molecules. Because we've got 3 times 10 and 2 times 2, so that's 34. But we also had a net gain of 2 in glycolysis and 2 produced in the Krebs cycle. So that's why overall from one glucose molecule you get 38 ATP. Whereas from anaerobic, we only get a net gain of 2 ATP in glycolysis. So there is a big difference there in how much ATP is produced in those 2 different reactions. So you do need to know this example as well, the rice plant adaptations, because rice plants have evolved several adaptations for growing in flooded or waterlogged conditions. So the erenchyma development, which is a specialised tissue with air-filled spaces in the roots and stems, this is to facilitate gas exchange, so oxygen and carbon dioxide fusing in and out between the roots and the surrounding water. It allows respiration when submerged by providing an oxygen pathway to the root tissues, and it reduces root weight, aiding buoyancy in those waterlogged conditions, so it will float. You also have ethanol fermentation in the roots, and that occurs under anaerobic conditions in waterlogged soils if there's limited oxygen. And that provides an alternative metabolic pathway for ATP production. That's going to help the rice plants to survive even in very, very low oxygen environments. And then lastly, we've got these resource allocations to the stems and the leaves. That's prioritising growth of aerial parts of the plant when it's submerged. And that is going to maintain photosynthesis and overall growth despite the adverse root conditions, and maximise exposure to sunlight and carbon dioxide for photosynthesis and carbohydrate production. So collectively, those adaptations enable rice plants to thrive in flooded or waterlogged environments, facilitating rice cultivation in high rainfall regions or irrigated paddy fields. So topic 13 is all about photosynthesis, and we start by looking at the structure of a chloroplast, which is where photosynthesis happens. So you've come across this already when learning about cells and cell organelles. So you should have some idea already about the thylakoid membranes, which we can see here are folded to make these disc-like stacks, which are the granum. And the thylakoid membranes, that is where you have lots of proteins embedded, which is going to be the site of chemiosmosis in the light-dependent reactions, which we're going to come on to. The stroma is this fluid-filled centre, which is the location of the light-independent reactions, and there's lots of enzymes in the stroma for catalysing those reactions, which we'll come on to later. And then we've got the inner and outer membrane. This is a double membrane-bound organelle, so you don't find them in prokaryotic cells because it is membrane-bound. So inside of the chloroplast there is chlorophyll, and chlorophyll is located in the photosystems, which are proteins embedded on the thylakoid membrane. And it's a mix of coloured proteins that absorb light. That's what chlorophyll is. There's about 5 closely related types of pigments. In reality there's lots, but you don't need to know all of them. Chlorophyll A is the most abundant, and the other pigments are listed below as well. So chlorophyll A is a pigment which is bluey-green in colour, found in all plants. Chlorophyll B is a yellowy-green colour. The carotenoids are orange, the xanthophylls are yellow, and the phaeophytins are grey. And these different pigments look different colours because they reflect different wavelengths of light, which also means they absorb different wavelengths of light. And this is why it's an advantage for plants to have different pigments, because that will maximise the amount of light energy that can be absorbed across the spectrum of the visible light wavelengths. And each plant has a different proportion of each pigment, which is why the leaves look slightly different colours. But that is the advantage, and we can see it here on this absorption graph against the wavelength. It's actually only showing chlorophyll A and B on this graph, but we can see even just those two different pigments, which are both shades of green, absorb wavelengths, or different wavelengths at different proportions. And if we were to add in the other pigments, we'd have an even bigger spectrum of the wavelengths where we'd have higher absorption. And ultimately, as I said, that means light, you're going to have more light energy being absorbed for photosynthesis. So chlorophyll B, xanthophylls and carotenoids, those are all embedded within the thylakoid membrane and form a light harvesting system. And by that we mean it's where light energy of different wavelengths is absorbed, and this energy is then transferred to the reaction centre. The reaction centre contains chlorophyll A and is where light-dependent reactions occur. So the light harvesting system and the reaction centre combined make up a photosystem. So back to looking at chlorophyll then. As I said, each pigment can absorb a different range of wavelengths of light within the visible light spectrum, but 500 to 550 nanometers is poorly absorbed by all pigments. So wavelengths within this range are reflected, and that's why leaves look green. The advantage we've already talked about, but it's now starting to link more specifically to the light-dependent stage of photoionization and photolysis. The pigments in chlorophyll can be isolated using chromatography, which is one of the practicals you're expected to know how to do. So for example, TLC chromatography, which this image here is from a TLC, which is thin layer chromatography. The pigments from the plant would be added at the origin line to your TLC paper. You would then place the paper in solvent. The pigments dissolve in that solvent, and depending on how soluble each pigment is in that solvent means how far up the TLC paper the pigment can travel. You can then calculate an RF value, which is a value that you can use to compare and identify pigments in chromatography. And the way you work out an RF value is using this formula below. So you'd have to measure with a ruler the distance moved by the pigment from the origin point to the centre of the point of the pigment. So looking at the distance the pigment moved. And we'd measure it from the centre because you can see here that the pigment is actually quite spread out. So measuring from the centre each time standardises the method, but also it's a way of working out the average distance moved. And then you divide that by the distance from origin to the solvent front. So measuring from this point to wherever the solvent travelled up to. And that's why when you take your TLC paper out of the solvent, you have to draw a line straight away to show where the solvent travelled to, because the solvent will evaporate and you won't be able to see that afterwards. So we're then going to move on to the actual reactions of photosynthesis. And it begins with the light-dependent reactions. And this involves the harvesting of energy from light. And the purpose of this stage is to use that light energy to split water and to create ATP and the reduced coenzyme NADP, which are then needed in the subsequent stages of the light-independent reactions. Now, the light-dependent reactions happen on the thylakoid membrane, or the grana, which is the stacks of those membranes. It involves 4 key steps, which we're going to go through. Non-cyclic photophosphorylation, cyclic or cyclic photophosphorylation, photolysis, and chemiosmosis. So we're going to start with the non-cyclic phosphorylation or photophosphorylation. This involves 2 photosystems, photosystem 1 and photosystem 2. Photosystem 2 is the first one to be used, which is confusing because it's called number 2, but it's the first one that's used, but that's just the way it is. And that photosystem has pigments within it to absorb light with a wavelength of 700 nanometers. Photosystem 1 is then used and it absorbs light with a wavelength of 680 nanometers. So here we can see those 2 photosystems embedded within the thylakoid membrane. We've got photosystem 2, photosystem 1. And on this side of the membrane, it's the stroma. On this side of the membrane, it is the lumen, so it's the space within the foldings of that thylakoid membrane. So light energy is absorbed. So the light shining from, it could be the sunlight, for example, is absorbed by the pigments within photosystem 2. And this causes the electrons within the reaction center of photosystem 2 to become excited, and they are then released. The electrons released from photosystem 2 and from photosystem 1, because it happens to both of these photosystems, move along an electron transport chain. That is what these proteins are here. And you don't need to know the names of these. You just need to know there are proteins embedded within the thylakoid membrane, and the electrons are going to be excited and released from the photosystem 1, photosystem 2, and then they pass along these electrons, which is the electron transport chain. This results in ATP production by ATP synthase over here, and that's chemiosmosis. And the electrons lost from photosystem 2 are replaced by electrons from photolysis, which we're going to go on to chemiosmosis and photolysis in the next couple of slides. The electrons lost from photosystem 1 are replaced by the electrons at the end of the electron transport chain from photosystem 2. So as the electrons are lost from photosystem 2 and pass along the electron transport chain, that is then picked up by photosystem 1. In comparison, the electrons that are lost from photosystem 1 and move along the electron transport chain, those are picked up by the coenzyme NADP, and that is then what reduces the NADP to NADPH, or reduced NADP. So the reduced NADP and the ATP that are made are used in the next stages of photosynthesis, which is the light-independent reactions. But there's still more to say on the light-dependent reactions first. Cyclic photophosphorylation is the next step, and this is happening simultaneously because as that light energy is absorbed by photosystem 1, we said that the electrons are excited, raise an energy level, and they're released, and they pass along the electron transport chain, and some of them are picked up by NADP to become reduced NADP. But some of them are instead recycled back into photosystem 1. And that transport of electrons still results in ATP production through chemiosmosis, which is a process we're going to come on to. Therefore, cyclic phosphorylation results in the production of ATP, but not the production of reduced NADP, because it's this cycling back round into photosystem 1 bypasses this step. The electrons are not being picked up by NADP, instead they're recycled back into photosystem 1. So cyclic photophosphorylation just results in ATP production, whereas non-cyclic photophosphorylation results in ATP and reduced NADP production. So the other stages then This is all happening simultaneously. Photolysis or photolysis, photo means light, lysis means splitting. So this is the stage where the light energy is absorbed by the chlorophyll, and that energy splits water into oxygen, electrons, and protons. The protons, the H+, is picked up by the NADP to form reduced NADPH. The electrons are going to be passing along the electron carrier proteins and the electron transport chain. The oxygen isn't used in photosynthesis, so it might be used in respiration, or it will just diffuse out of the stomata and leaf. So chemiosmosis then, this is the last bit just to look through. And the diagram, you can see here again the effects of non-cyclic photophosphorylation and also cyclic phosphorylation, and how we get the reduced NADP and ATP production. But the main thing we're focusing on here is how we get the ATP production, because we've not explained that part yet. So the electrons that gained energy and left the chlorophyll in photosystem 2, and it would happen in photosystem 1 as well, move along those proteins in the electron transport chain. As they move along these proteins, they release energy. And that energy is used to pump protons from the stroma into the thylakoid lumen, so across the membrane. This results in an electrochemical gradient, because you'll have lots of protons in the thylakoid lumen compared to in the stroma. And because of this electrochemical gradient, those protons will now move by facilitated diffusion back into the stroma down their concentration gradients. But the only way they can do that is through the enzyme ATP synthase, because protons, they have a charge, they can't dissolve and diffuse through by simple diffusion in the thylakoid membrane, and the only protein that they are able to bind to and travel through is ATP synthase. So as they move through ATP synthase, that catalyzes or activates ATP synthase to catalyze ADP and PI, inorganic phosphate, joining together in a condensation reaction to make ATP. So that's how we make the ATP. Those protons that have now moved back across to the stroma, some of those protons are then going to be picked up by NADP, which is also picking up the electrons at the end of the electron transport chain, and that is how we create the reduced NADP, or in other words, NADPH. So that's how we get the ATP and the reduced NADP, both of which are needed for the light independent reactions, which is a second stage of photosynthesis, which is also known as the Calvin cycle. Now, this stage happens in the stroma, and it involves the enzyme Ribisco, and it's a temperature sensitive stage because of the enzymes. Now, the Calvin cycle uses carbon dioxide as well as the two products from the light dependent reaction, reduced NADP and ATP. And it creates the hexose sugars, or in other words, the organic substances that the plant is then going to use to make carbohydrates, amino acids, lipids, all the organic substances that it needs. And this stage involves ATP from the light-dependent reactions being hydrolyzed to provide energy for the reactions, and the reduced NADP donating the hydrogen atoms, which is a proton and an electron, to reduce molecules, for example, GP in the cycle. But let's have a look at it step by step. So we begin here. Carbon dioxide is reacting with ribulose bisphosphate, or RUBP, and we've got a 5-carbon combining with a 1-carbon compound, and that creates a 6-carbon compound. You don't actually need to know that step, but the 6-carbon compound splits into 2 molecules of GP. And this is a step that requires the enzyme Ribisco. Those 2 molecules of GP are both reduced to 2 molecules of triose phosphate, or here it's summarized as TP. And that is going to be reduction because the hydrogen, which is a proton and an electron from reduced NADP, is picked up by those 2 GP molecules. And this stage also requires energy from ATP. Now, once we've got those 2 molecules of TP, 1 carbon from the 2 molecules of TP is removed from the cycle each turn of the cycle. Now, only 1 is, which means we're left with 5 carbons, And those 5 carbons are then going to be regenerated, joined together to make RUBP again. And that regeneration stage again requires ATP. It's the energy from ATP is needed to join together the 3 and the 2 carbon compound. And that 1 carbon that is removed each turn is going to go towards making the useful organic substances. So it takes 6 turns of the Calvin cycle before you have 6 carbons to create a hexose sugar, such as glucose. And whilst glucose is the product, that monosaccharide could join together with fructose to make sucrose, or many glucose molecules could join together in condensation reactions to make polysaccharides, such as cellulose and starch. Or the hexose sugars could be converted into glycerol, which is a 3-carbon sugar, and that goes towards making the lipids, or you could even add nitrogen to it to start to make an amino acid. So there are limiting factors that affect the rate of photosynthesis, and that is temperature, carbon dioxide concentration, and the light intensity. So with the light intensity, as you increase light intensity, the rate of photosynthesis increases at the lower light intensities. And that's because the more light there is, you'll have more photolysis of water, and you'll have more electrons being excited and released in cyclic and non-cyclic photophosphorylation. And therefore you ultimately get more reduced NADP and ATP used in the Calvin cycle. It does, though, level off or plateau at the high light intensities, and at that point there would be another factor limiting the rate. So you've already got to your optimum light intensity, so at this point it must be there's not enough carbon dioxide for the Calvin cycle, or it might be the temperature is not high enough to reach the optimum temperature for the enzymes. Carbon dioxide concentration, we have the same curve. At the lower concentrations of carbon dioxide, we see that it is limiting the rate because we've got this directly proportional relationship. And that would be because carbon dioxide is required to react with ribulose bisphosphate or RUBP in the Calvin cycle. However, again, we get to the point where the rate of reaction plateaus, and at that point there must be another factor limiting the rate of photosynthesis, and that would either be the light intensity or temperature. Temperature, the shape of the graph is different because this is linked to enzymes. So you might have a steep curve like this, or it might be more gradual and then a drop off, because as you increase the temperature, the enzymes will be gaining, and also the substrates will be gaining more kinetic energy, so you'd get more successful collisions between the enzyme and the substrate, and therefore the reaction happens faster. But beyond the optimum temperature, the enzyme starts to denature, and that's why the rate of reaction doesn't plateau, it actually decreases, because if the enzymes are denaturing, then we aren't catalyzing the reaction. Now, for maximum photosynthesis, in agriculture, they'll need to try and find a balance between the optimum amount of light intensity, carbon dioxide concentration, and temperature, but offset that with how much it costs to have that heating, to have that extra lighting, compared to how much extra profit they're getting from how much the plants are actually growing. So you do have to consider how cost effective it is to potentially pay for paraffin to burn it, which would release carbon dioxide but also increase the temperature, and to pay for artificial lighting. So let's get into topic 14, which is on homeostasis. And we're starting with homeostasis in mammals. So multicellular organisms require a communication system, and animals and plants need this to respond to changes in their internal and external environment, and to coordinate the activities of different organs to help them to survive. And these communication systems are the nervous and hormonal systems, which together coordinate the activities of the whole organism. So through chemical signalling, cells are able to communicate with adjacent cells, but also cells that are really far away from them. The cells that the chemicals will affect are called target cells, and we're going to be looking at particular target cells in this video linked to controlling the water potential of the blood and the blood glucose concentration of the blood. Some chemical signals affect adjacent cells only, such as the release of neurotransmitter at the synapse, but that's in the nerve system video. Other chemicals, which is what we're focusing on in this video, they will have signals that transfer between distant cells and a large distance across the organism. So for example, insulin, which is released by pancreatic cells, is transported in the blood to the liver, where it acts on the target cells, which are the liver cells, to regulate blood glucose concentration. So this video is all about homeostasis, and the definition of that is the maintenance of a constant internal environment via physiological control systems. So essentially, it's making sure that the conditions inside of you remain within set limits, and if they get too high or too low, then systems are put in place to bring it back within the set limits. And the things that are controlled, or the conditions that are controlled, are temperature, the pH of your blood, the amount of glucose in your blood, and blood water potential. But we're only going to be focusing on the blood glucose concentration and the blood water potential. And homeostasis involves both negative and positive feedback loops. The two examples that we're looking at are focusing on negative feedback loops, which is the concept that if a condition is going to move outside of the set limits, mechanisms are put in place to bring it back down or back up into that set limit. And that's what we can see here with our negative feedback loop. If we get this deviation from the set limit, and that is detected by the body, so a receptor will detect it, mechanisms are put in place to restore those conditions back within the set limits. So we can see here if the body temperature exceeds 37 degrees C, which is outside of the normal set limits, it's going to be detected by cells in the nervous system. That is then going to result in mechanisms happening such as sweating, which will then result in cooling, which brings your body temperature back to the normal limit. And this negative feedback involves the nervous system because you have to have receptors to detect the change. And it often involves hormones too, because those are chemicals that can cause the change to occur. So we're going to look at controlling the water potential of the blood first, and this involves the kidney because this is where the blood is filtered and excess water is removed. So this is also responsible for removing nitrogenous waste, not just excess water. And osmoregulation is the process or the term for controlling the water potential of the blood. And if we have a look at the kidney, we can see here the renal artery and the renal vein. The renal artery supplies the kidney with blood that needs to be filtered, and the renal vein carries that filtered blood away from the kidney. The kidneys are made up of three distinct layers. We have the cortex, the medulla, and the pelvis. So looking at these in a bit more detail then, those are three layers that we just talked about. We've got the cortex, medulla and pelvis. The cortex is a dark outer layer, so we can see here the cortex, and that contains capillary networks carrying blood from the renal arteries to the nephrons. The medulla, which is this section here, that contains the nephrons. And then lastly, we have the pelvis, which we can see here. That collects urine before it travels to the ureter. Now there's also this fibrous capsule. That is a dense connective tissue layer enveloping the kidney. It provides support and protection. It adheres, which means sticks, to the renal cortex. And it's going to also maintain the shape and structure. So we can see here that fibrous capsule. It also acts as a barrier against mechanical stress and trauma, and it can isolate internal structures from potential infection or damage as well. So it's isolating the kidney. The renal blood supply we've briefly talked about already, because we said we've got the renal artery, which supplies the kidney with blood to be filtered, and the renal vein, which carries the filtered blood away. And then lastly, the nephrons, which we're going to be talking about in more detail. The nephrons are within the medulla. You actually have millions in each kidney, and these are the structures where the blood is filtered and the useful substances are reabsorbed back into the blood through capillaries. And whatever isn't reabsorbed forms the filtrate, which goes on to forming the urine. So here we can see one of the nephrons inside of the medulla. And we're going to talk about what happens at each point of the nephron and what the different structures are. So we begin with the Bowman's capsule or the renal capsule, which is this bowl-like structure. that is where you have lots of blood vessels inside of it. So the capillaries that make up the glomerulus, that is where ultrafiltration occurs, which is where you have a really high blood pressure forcing out the water and small molecules such as glucose and urea. And that forces out those molecules into the renal capsule, and that now forms the glomerular filtrate. That filtrate then passes through the proximal convoluted tubule, and that is where glucose is going to be reabsorbed, but also some water gets reabsorbed at that point. The filtrate then moves down the descending loop of Henle and then back up the ascending loop of Henle. And it's in the loop of Henle where we have sodium ions being actively transported out, which will then be reabsorbed, and also lots of water is going to move out by osmosis. Then the filtrate moves through the distal convoluted tubule and the collecting duct, and at those points we have even more water being reabsorbed back into the blood from the filtrate. But if we go through each of those steps in more detail. The ultrafiltration, and this is what we said happens at the renal capsule or the Bowman's capsule. Blood enters the glomerulus, the network of capillaries within the Bowman's capsule. And because the blood enters through a blood vessel known as the afferent arterioles, which then splits into lots of smaller capillaries, which therefore have a narrower lumen, it results in this high hydrostatic pressure. So you've now got the same volume of blood, but in a smaller space. And that hydrostatic pressure is what causes the water and small molecules such as glucose, mineral ions, and urea to be forced out and form the glomerular filtrate. Now, this is showing you how you have that filtration occurring, because this is showing you one of the capillaries that make up the glomerulus, and capillaries are only one cell thick. So the gaps between those cells are small enough that you won't have large molecules being forced out, like red blood cells and large proteins, but small molecules can fit between those gaps. And that's what these tiny circles are meant to represent, the gaps between the cells in the wall of the capillary. Now, lining that capillary, you can see this blue layer, which is the basement membrane. And that is another layer that filters. And then surrounding those, you can see these yellowy beige coloured cells, which are known as podocytes. And that creates another layer that the liquid has to pass through. So we've got those three filter layers. We've got, once it's forced out of the holes or the gaps or the fenestrations in the capillary, it then has to pass through the basement membrane. That acts like a sieve. And then after that, it has to pass through the gaps between the cells, which are the podocyte. That filtrate will then flow through to the proximal convoluted tubule. And this is where we have most of the filtrate reabsorbed back into the blood, just leaving behind the urea, excess mineral ions, and some of the excess water in the filtrate. The main things that are happening here, though, are the glucose is going to be actively transported. And this is an example of co-transport, much like you would have learned in digestion, the co-transport of sodium ions and glucose from the lumen into the cells. That is what's happening here as well. So sodium ions are going to be actively transported out of the epithelial cells in the proximal convoluted tubule into the blood, into the capillary. That lowers the concentration of sodium ions in the epithelial cells compared to the lumen, which is the hollow tube center. And therefore sodium ions will move from the lumen into those epithelial cells lining the PCT. But as those sodium ions move in, down their concentration gradients, they're actually co-transported with glucose. So that's how glucose will enter the epithelial cells of the proximal convoluted tubule. And then the glucose can move from the epithelial cells into the blood by facilitated diffusion. And this is how all of the glucose is reabsorbed, or almost all of the glucose is reabsorbed from the filtrate back into the blood. And that is really important because we don't want to be losing glucose in our urine, because glucose is needed for respiration. And if you do have excess glucose in your blood, that can be stored as glycogen, which we're coming on to later in this video. Now, some water does also get reabsorbed at this stage, and that's because as those sodium ions are actively transported from the epithelial cells into the blood, that lowers the water potential of the blood, so water can move by osmosis into the blood. But most of the water reabsorption is going to be happening in the loop of Henle. And what we have happening here is that filtrate passes from the proximal convoluted tubule into the loop of Henle down the descending limb. Now in this image, they're actually looped over or crossing over each other. The filtrate does go down the descending limb first and then up the ascending limb. And as the filtrate moves down the descending limb, this is where you're going to have water being reabsorbed. But that's only possible because of what happens in the ascending limb. So let's have a look at what happens in the ascending limb. You have mitochondria, you have lots of mitochondria in the walls of the cells lining the ascending limb. And that is so that they can provide ATP for energy to actively transport ions that are in the filtrate as it moves up the ascending limb. And those ions are transported out of the ascending loop of Henle to the surrounding space, which is known as the interstitial space. Because you now have lots of sodium ions, and also you have some chloride ions as well that are actively transported out, but lots of ions in that interstitial space, that lowers the water potential of the medulla in that interstitial space. Because the water potential is lower, that then means water will move out of the descending limb by osmosis. And the reason the water isn't moving out of the ascending limb is the ascending limb has much thicker walls which are impermeable to water. So water can only move out of the descending limb, and it moves out by osmosis because of that more negative water potential. And once it moves out of the descending limb, it will then move into the blood capillaries, and that's why we say it's reabsorbed, it's going back into the blood. At the base of the ascending limb, some sodium ions are transported out by facilitated diffusion, as there's now very dilute solution there because of all the water that is also moving out by osmosis. So when the filtrate then moves to the distal convoluted tubule, or DCT, and the collecting duct, even more water is reabsorbed back into the blood. And that is because of all the sodium ions that are being actively transported out of the ascending limb of the loop of Henle, lowering the water potential. So that when the filtrate reaches the distal convoluted tubule, that filtrate is very dilute because lots of the ions have been removed. And that filtrate moves into the DCT and the collecting duct, and at that point, the interstitial space in the medulla is very concentrated and you've got a very dilute solution. Therefore, even more water diffuses out of the DCT and the collecting duct. And whatever remains in terms of the filtrate in the collecting duct then goes on to form the urine. So that is how the nephron filters the blood, but we need to know how this links to homeostasis, meaning what happens when there's now too much water or too little water in the blood. And this is where we see the involvement of the nervous system. And it involves the hypothalamus and the posterior pituitary gland in particular. The hypothalamus is where you find the osmoreceptors, which are the cells that detect changes in the water potential of the blood. If the water potential of the blood is too low, water will leave those osmoreceptors by osmosis and it causes them to shrivel. And this stimulates the hypothalamus to produce more of the hormone ADH. If the water potential of the blood is too high, water will then move into the osmoreceptors by osmosis, and this stimulates the hypothalamus to produce less ADH. So the hypothalamus is where this hormone ADH is produced. The ADH then moves from the hypothalamus into the posterior pituitary gland, and it's the posterior pituitary gland which releases the ADH. And the ADH will then travel through the blood, bind to the target cells and the target organ, which are the kidney. So what is ADH then? ADH is antidiuretic hormone, and once it's been released by that posterior pituitary gland, transported in the blood, it will bind to complementary receptors, which are only located on the cell surface membranes of target cells, which are the kidney cells, specifically the cells lining the distal convoluted tubule and the collecting duct. So ADH is only going to affect those cells. When it binds, it activates adenyl cyclase to make cyclic AMP or CAMP. And that activates an enzyme which causes vesicles within the cells that contain aquaporins to fuse with the membrane. And aquaporins are channel proteins embedded within the cell surface membrane of the DCT and collecting duct, which allow water to be transported across the membrane. So if you have more aquaporins embedded within the membrane, that means the cell surface membrane is now more permeable to water, so more water can leave the distal convoluted tubule and collecting duct to be reabsorbed back into the blood. So the more ADH that is released, the more aquaporins there are in the membrane, and therefore the more water gets reabsorbed back into the blood. So that's what we can see happening here, and it results in lower volumes of urine being created, which are more concentrated. So we can see here, these are the vesicles that contain the aquaporins, which are being represented by these pink circles. Those vesicles then move and fuse with the cell surface membrane, so you get lots more aquaporins being embedded, and therefore more water can travel through the aquaporins back into the blood. So if we have a look then at how this helps to regulate the water potential, if the water potential of the blood increases, so meaning you've got too much water in your blood, so maybe you've had a lot to drink in terms of water, that is going to be detected by the osmoreceptors in the hypothalamus. That causes the hypothalamus to produce less ADH, and therefore the pituitary gland is going to release less ADH. That means the DCT and collecting duct walls become less permeable to water because you're not going to have as many aquaporins being embedded. Less water is reabsorbed into the blood, and more water is therefore remaining in the filtrate to form the urine. So as a result, you have larger volumes of urine and it's more dilute, meaning more water in it. So you're removing that excess water, and that brings the water potential back within the normal set limits. The opposite then is if the water potential of the blood decreases, meaning you don't have enough water in the blood. So maybe you haven't drunk enough water, maybe you've been sweating a lot. That will be detected by the osmoreceptors in the hypothalamus. That causes the hypothalamus to produce more ADH, and therefore the posterior pituitary gland releases more ADH. That is released into the blood. That causes the DCT, the distal convoluted tubule, and the collecting duct walls to become more permeable to water. More water is reabsorbed back into the blood, and therefore you have smaller volumes of urine and it's more concentrated. And as a result, your water potential goes back to its normal set limit. Next, then we're going to have a look at the control of blood glucose concentrations, and this involves the pancreas. And the pancreas is a gland behind the stomach. It releases hormones to control blood glucose levels, and therefore it's a functioning endocrine gland. It does actually also secrete enzymes for digestion, which means it's an exocrine gland as well. Most of the pancreas is made up of exocrine tissue, so releasing those enzymes, amylases, proteases and lipases, but there is a small region of endocrine glands which are going to make up the islets of Langerhans. And these are the parts of interest for homeostasis. The islets of Langerhans are made up of alpha cells, which are going to be secreting glucagon, and beta cells that secrete insulin. And that is what we can see here. Whilst most of the cells here are exocrine, secreting enzyme, we can see here in blue those islets of Langerhans, which are cells that will be secreting the hormones linked to blood glucose concentration control. So the blood glucose concentration will increase following the ingestion of food or drink containing carbohydrates. And you do have to emphasize containing carbohydrates, not just saying after you've eaten or drunk. It's specifically containing eaten or drunk anything that contains carbohydrates. And it's going to decrease if you haven't eaten anything that contains carbohydrates in a while, but also following exercise, because exercise requires lots of ATP. ATP is produced from respiration, and glucose is a respiratory substrate for that. So it'd be using up lots of glucose from the blood. So the key structures and hormones then are the pancreas, because that is where you have your islets of Langerhans, and that contains the receptors that detect the changes, but also the effectors, which will release the hormones insulin and glucagon. We can have a look at the role of these hormones as well. Insulin is the hormone that's released when blood glucose levels are too high, and it will cause a decrease in the blood glucose levels. Glucagon is released when the glucose levels are too low. I always say think of glucagon is released when the glucose is gone, and that will cause an increase in the blood glucose levels. Adrenaline isn't actually linked to homeostasis, but it is a hormone that will cause an increase in blood glucose levels, and it's released when you are anticipating danger, but it has the same effect as glucagon. So in terms of homeostasis, your normal blood glucose levels will increase following a carbohydrate-rich meal or drink. That increase is detected by the beta cells in the islets of Langerhans and the pancreas. The beta cells will release the hormone insulin. We're going to look at in more detail what insulin does, but insulin causes liver cells to become more permeable to glucose. Therefore, glucose can move from the blood into the liver cells. And once that glucose is inside of the liver cells, insulin also activates enzymes to convert glucose to insoluble glycogen stores. Therefore, glucose has been removed from the blood, stored as glycogen in cells, and we've gone back to our normal blood glucose limits. The opposite then is if the blood glucose levels decrease. So maybe you've done a lot of exercise or you haven't eaten anything containing carbohydrates for quite a period of time. That decrease will be detected by the alpha cells in the islets of Langerhans and the pancreas. The alpha cells will then release the hormone glucagon. And glucagon, we're going to look at how that results in the glycogen being hydrolyzed back into glucose, being released into the blood through the second messenger model. But ultimately, that is what happens. And therefore, glucose returns to the blood and it increases your blood glucose levels. So let's have a look at how you can measure the blood glucose levels. This could be using test strips and biosensors. And you might want to measure your blood glucose levels to test for diabetes, for example. And these are commonly used to measure the concentration of glucose in blood and urine. And these devices use enzyme reactions, and this is how they would work. So we've got the comparison of the test strips compared to the biosensors. And you can see what is on those strips versus the sensor, the enzymes used, the reactions involved, how it detects it, what's being measured, the output, the advantages of the two, and what it's being used for. So test strips are commonly used for quick on-the-spot glucose testing, whereas biosensors are used in more advanced glucose monitoring devices, including continuous glucose monitoring. So you can pause the video, read through this information in your own time, or you can pause, take a screenshot, print it out, and then you've got that table to go in your notes. But if we go back to then the effect of the hormones, and we're going to start with insulin. Insulin, we said, is secreted by the beta cells in the islets of Langerhans when blood glucose levels are too high, so following a carbohydrate-rich meal. And it results in the decrease in blood glucose in the following ways. First of all, the insulin, once it's secreted by those islets of Langerhans, it's transported in the blood and it will bind to complementary shaped receptors, which are embedded in the cell surface membrane of the target cells and the target organs, which is mainly the liver cells. When that insulin binds to the receptors, it changes the tertiary structure of glucose channel proteins, and that results in them being wider, so more glucose can be transported from the blood into the cells by facilitated diffusion. Not only that, when the insulin binds, it results in more protein channels being embedded within the cell surface membrane. So again, more glucose is going to be absorbed from the blood into the liver cells. Now, all of that glucose that's been removed from the blood and into the cells has to be converted into glycogen because glucose is soluble. Therefore, it would lower the water potential of the cytoplasm of the cells, causing water to move into the cell by osmosis and the cell would burst. Whereas glycogen is insoluble. So that's why the insulin also activates enzymes to convert glucose to insoluble glycogen. And that's known as glycogenesis, making glycogen. Genesis means to make, glyco means glycogen. Now, this is actually just showing you the concept of how you get more channel proteins embedded within the membrane. So insulin which is a hormone, is binding to the insulin receptors on the target cell of the liver. That causes a cascade of reactions here, which you don't need to know. But essentially, you have a range of different chemicals being released, which causes vesicles within the liver cells, which contain the aquaporins, to move up to the cell surface membrane, fuse and embed more glucose channel protein. And because you then have more channel proteins embedded, more glucose can be absorbed into the cells. Then if we have a look at the action of glucagon, alpha cells in the islets of Langerhans detect when the blood glucose is too low and they will secrete glucagon. So when the glucose is gone, secrete glucagon. And this causes an increase in blood glucose in the following ways. Again, it's transported in the blood, attaches to complementary shaped glucagon receptors on the liver cells. But when the glucagon binds, it causes a protein to be activated into adenyl or adenylate cyclase and to convert ATP into cyclic AMP. Cyclic AMP activates an enzyme, which is a protein kinase, and that enzyme can hydrolyze glycogen into glucose. And that glucose is then released back into the blood. It's also responsible for activating enzymes involved in converting glycerol from lipids and amino acids from proteins into glucose. And that step is gluconeogenesis. And that will only occur if you haven't eaten anything containing carbohydrates for a prolonged period of time and your glycogen stores have been used up. So let's have a look at this process in more detail. It's known as the second messenger model. We've got the glucagon binding to the receptors. We said that causes a change in shape to adenyl cyclase, which is an enzyme. And that change in shape of the enzyme activates the enzyme, so it can catalyze the reaction of converting ATP into cyclic AMP. This is known as the second messenger, and we call it a messenger because it's able to cause a change. So glucagon was the first messenger, because when it bound, it caused this change here. The cyclic AMP can cause the change of inactive protein kinase to active protein kinase, and active protein kinase can catalyze the hydrolysis of glycogen into glucose. And that glucose will then be returned to the blood. So here are our key G words that have come up, and we have glycogenesis. Anytime you see glyco, that means glycogen. Genesis means to create. So glycogenesis is when we are creating glycogen from glucose. Glycogenolysis, we can even see the word glycogen written here, and lysis means to split. So glycogenolysis is the splitting or hydrolysis of glycogen back into glucose. Gluconeogenesis, gluco means glucose, neo means new, genesis means to make. So gluconeogenesis is making new glucose from non-carbohydrate sources. So for example, the amino acids from proteins and the glycerols from lipids. So we then move on to homeostasis in plants. Stomata, which are vital for maintaining a constant internal environment in plants, respond to environmental changes by opening and closing. And this regulation of stomatal aperture, meaning how open or closed they are, is necessary for carbon dioxide uptake whilst reducing water loss by evaporation. So this is going to maintain a constant internal environment for the plants. It involves responding to environmental changes, so the opening and closing is the response, and it's going to help balance carbon dioxide uptake with water loss. So stomata will open if there is a high light intensity. Also, it's going to be triggered by low carbon dioxide within the leaf, because if the stomata is open, that means more carbon dioxide can diffuse into the leaf. And it will lead to more water loss, though, through transpiration. So if the stomata are open, you do get more carbon dioxide diffusing in, but you'll have more water vapour evaporating out. The stomata will close if it's darker. It's also going to be triggered by high carbon dioxide concentrations in the plants, triggered by low humidity and high temperature because you want to reduce water loss under those water stress conditions, because when it closes, it reduces transpiration and therefore it conserves water. So how that happens then? The guard cells are the cells that bend and change shape to create the stomata. and they are going to have these thickened celled walls with the cellulose fibres, the high density of chloroplasts and mitochondria, and they open when they gain water and become turgid. So the stomata opens when the guard cells bend. They're going to bend like this when lots of water moves in, causing the cell to become so full of water and turgid that they bend and create that opening. They will close when there isn't as much water inside of them, so the cells become flaccid. That causes them to close or move close together, so the stomata closes. So how that happens then, the opening involves the active transport of hydrogen ions out of the guard cells, causing potassium ions to enter and decrease the water potential. Therefore, water enters the cells via osmosis, increasing that turgor pressure and leading to the stomata pore dilation. Or in other words, it opens. The closing is triggered by factors such as darkness and water stress, and it involves the stopping of the hydrogen ion pump proteins and the potassium ion efflux and water efflux. Also, the hormone ABA plays a pivotal role in stomatal closure during water stress by stimulating calcium ion influx, which activates channel proteins to induce potassium ion efflux. And that will lead to guard cells becoming flaccid and the stomata closing. So ABA acts as a stress hormone, coordinating plant responses to challenging environmental conditions. Topic 15 then, and it's all to do with control and coordination. So in chapter 14, we saw how homeostasis occurred to help regulate blood glucose levels and water potential, and you can actually have a look at that previous video as well. But in this video, it's going through how you can also have coordination and control using the nervous system. And with the nervous system, it's going to be far more rapid rather than gradual, which is what the hormonal response is. So insulin, glucagon, ADHD and adrenaline are examples of hormones which act as signaling molecules released into the bloodstream, and they travel long distance from where they're produced to their target organs. And these hormones are produced within endocrine glands, which basically means a gland that produces a hormone, and groups of cells responsible for secreting one or more substance directly to the bloodstream. Endocrine glands lack ducts, and the endocrine system encompasses all those ductless glands within the body, including the pituitary gland, which we can see at the top just here within the brain. So if we have a look at the nervous system compared to the endocrine system, I've put together this summary table for you so you can screenshot it, print it out and put it into your notes, or maybe turn it into flashcards. But you need to know the comparisons between the mode of communication, so how that information is delivered, electrical impulse versus hormones, the speed, duration of the effects, the target tissues, response time, adaptation to stimuli, regulation of functions, organs involved, mechanisms of actions, and feedback regulation. So these are all just key facts to know. So I'm not going to talk through all of this. You can screenshot and keep that in your notes. So moving on then, just focusing on the nervous system. The nervous system is made up of billions of neurons. These can be categorized into three main groups: sensory neurons, relay neurons, and motor neurons. And these three specialized cells do still have some features in common. So they all have a cell body, which contains the organelles that you find in a typical animal cell. So for example, the nucleus and the mitochondria. They have dendrons, which carry the action potentials to surrounding cells. And they have an axon, which is a conductive long fibre that carries the nervous impulse along the neuron. So if we then have a look at the actual structure we can see, so we've talked about some of those. Here's our cell body. We've got the nucleus inside of that cell body. This part is the axon, and we can see the dendrons branching out here as well. Myelinated neurons have Schwann cells as well, which is shown here in yellow, wrapping around the axon. And they wrap around to form what's known as the myelin sheath. And this is a lipid, and therefore it doesn't allow charged ions to pass through it. And in that way, it insulates and prevents the electrical impulse from travelling the entire length of the axon. Instead has to jump from node to node, which is known as the node of Ranvier, and that speeds up the conductance. So that's what we've got here, that those gaps are called the nodes of Ranvier, and we get that action potential jumping from node to node, which is known as saltatory conduction, and that is what speeds up how quickly that impulse can travel. So if we have a look at a sensory neuron, these are the neurons responsible for carrying electrical impulses from the sensory receptor cell to the relay neuron, sometimes also to the motor neuron and the brain. They have a long dendron which carries the impulse from the sensory receptor to the cell body, which is what we can see here, and there is the cell body, and then an axon to carry the impulse from the cell body to the next neuron. The intermediate neuron, sometimes known as the relay neuron, these neurons carry impulses between the sensory and motor neurons, and they have multiple short axons, which we can see here, and also dendrons branching off each of those. And then finally, the motor neuron, these carry impulses from the relay or sensory neurons to the effector, and the effector is either a muscle or gland, that is the part that carries out the response. And these have one long axon and multiple short dendrons. A sensory receptor then, these are cells that detect a stimulus. That is what a receptor is. And these cells are transducers, meaning they convert different types of stimuli into electrical impulses. There are different types of sensory receptors, and each type can detect a different stimulus. And here is your table summarizing the different types of sensory receptors and the stimulus that they detect. So photoreceptors, photo meaning light, they detect light, and that would be your rod and cone cells in your retina of the eye. Thermoreceptors, thermo means temperature, so they can detect changes in temperature or heat energy. Mechanoreceptors, so the Pacinian corpuscle and the sinus corpuscle as well, detect the pressure. Taste, smell and balance as well. So taste and smell are chemical, balance is movement. Next time we move on to the actual changes that happen in a response. And when there isn't a stimulus, your neurons would be in resting potential state. So that's what we're going to go through first. When a neuron is not conducting an impulse, meaning there's no stimulus, there is still a difference between the electrical charge inside and outside of the neuron, and that is known as the resting potential. You actually have more positive ions, which are the sodium ions and potassium ions, outside of the neuron compared to inside, and therefore the inside of the neuron is comparatively negative at minus 70 millivolts. So this graph here is showing you an action potential, which is what happens when there is a stimulus, but this section here of the graph, this is before there is any stimulus, and we can see that the voltage, which is our potential difference, is remaining at minus 70 millivolts. So the way that that occurs then is there is a sodium ion, potassium ion pump, and this stage requires ATP for active transport. And this co-transporter pump will pump 2 potassium ions into the axon of your neuron and 3 potassium ions outside of the cell. That creates an electrical chemical gradient because we'll then have more potassium ions inside of the cell and more sodium ions outside of the cell. And as a result of those electrochemical gradients, potassium ions will diffuse out of the cell through these channel proteins, and sodium ions will diffuse into the cell through their channel proteins. However, there are more potassium ion channels than there are sodium ion channels, so we say that the membrane is more permeable to potassium ions. Also, the potassium ions are permanently open, and some of the sodium ion channels are not permanently open. So as a result, you get more potassium ions diffusing out than you do sodium ions diffusing in. And as a result, you have more positive ions outside compared to inside. And that is how we get this minus 70 millivolts, or the resting potential. So if there is a stimulus, that can result in an action potential. And this is when the neuron's voltage, or potential difference, increases beyond a set point from that resting potential. So our resting potential is minus 70 millivolts. But if we actually get a change that brings the voltage up to the threshold point of minus 55 millivolts, then you will get an action potential occurring. So an increase in voltage, also known as depolarization, is due to the neuron membrane becoming more permeable to sodium ions, meaning more sodium ions are going to diffuse into the axon. And because those have a positive charge, that will bring that voltage to become more and more positive. So we go from minus 70 up to minus 55, which is more positive. Once an action potential is generated, it moves along the axon like a Mexican wave. So you'd have to generate that action potential at this point on the axon. That would then trigger an action potential at this node of Ranvier, and then this one, this one, this one, this one. And that's what we mean by the movement being like a Mexican wave. So let's go through then how an action potential is generated. We've got our resting potential here, where we have got those voltage-gated sodium ion channels closed. What that means is these are sodium ion channels that sodium ions can diffuse from the outside to the inside of the cell. However, those channels only open when there is a particular voltage, whereas we have some potassium ion channels which are permanently opened. So that's what's happening at our minus 70 millivolts for the resting potential. But if there is a stimulus, that stimulus causes some of the voltage-gated sodium ion channels to open. And as a result, sodium ions are going to diffuse in. And that causes the voltage to become more positive. Now, if you have a large enough stimulus, that will cause enough sodium ion channels to open to reach the minus 55 millivolt threshold. If it's only a small stimulus, that won't cause enough of those voltage-gated sodium ion channels to open, so you won't actually get an increase in the voltage to the threshold of minus 55 millivolts, and therefore you wouldn't respond to the stimulus. And that's actually really useful because you don't want to respond to every slight change because that would overwhelm your senses. So you're only going to respond if the pressure or the sound or the smell is strong enough that it does deem worthy of responding to, so that it would actually protect you from potential harm. Now, when you do get to that threshold point of minus 55 millivolts, that causes even more voltage-gated sodium ion channels to open, and therefore you get even more sodium ions diffusing in, making that voltage now become positive, and it peaks at plus 40 millivolts. Now, when you get to that point, that then causes those voltage-gated sodium ion channels to close, but we still have potassium ion channels that are permanently open. So we now have positive ions being diffusing out, so they're going to leave the axon. So we then start to become more negative again, which is known as repolarization. And at this point, we have even more potassium ion channels open, so even more potassium ions are diffusing out, which actually results in this overshoot where the voltage becomes even more negative than the resting potential. And we call this stage hyperpolarization, where the voltage can go to minus 80 millivolts. And this is known as the refractory period as well. During this point, these voltage-gated sodium ion channels are temporarily unable to open. So you get a very short period of time where a new action potential wouldn't be able to be generated. And we're going to talk about why that is an advantage. But for now, that is just showing you how that depolarization results in this action potential, and then how that afterwards results in repolarization going into that refractory period. But then, because of that sodium ion, potassium ion pump that we talked about in maintaining the resting potential, it does bring that voltage back up to the minus 70 millivolts of resting. So this is just what I meant by the Mexican wave. That action potential is generated at this point on the axon. That then jumps to the next node of Ranvier, the next one, and that will happen all the way along the axon until it reaches the end of that motor neuron, and then we get to a synapse. So the all or nothing principle is this concept that if the depolarization does not exceed that threshold of minus 55 millivolts, then you will not get an action potential. So nothing happens. That's the nothing part of all or nothing. Any stimulus that does trigger depolarization, meaning it has reached that minus 55 millivolt threshold, the maximum voltage is always the same at plus 40 millivolts. So a bigger stimulus doesn't result in a greater peak, a greater voltage. It's always going to be a maximum of plus 40 millivolts because of the set number of channel proteins present and the number of ions present. But what does vary is the frequency at which those action potentials are going to be generated. So the larger the stimulus, the quicker the frequency, or the higher the frequency. So this is what we mean by the all or nothing principle. If you don't reach that minus 55 millivolts, then nothing will happen. You won't get an action potential. But if you do, then you'll always get an action potential, and it will always peak at that same voltage. So it's important, as this makes sure that animals only respond to a large enough stimuli. And this is what I was talking about when we mentioned the threshold potential earlier. You have to have a large enough stimulus to reach that threshold, and therefore you're only going to respond to large enough stimuli and not every slight change in your environment, because that would overwhelm your senses. So a bit more about the refractory period. After an action potential has been generated, the membrane enters that refractory period when it can't be stimulated again. And that's because those sodium ion channels are recovering and they can't be opened. So that is important because it ensures that discrete impulses are produced. An action potential cannot be generated immediately after another, and that is why it makes sure that each impulse is separate. It also ensures that action potentials only travel forwards in one direction. It stops the action potential from spreading out and going backwards. And if it did go backwards along the axon, that would prevent it causing a response, because it wouldn't actually end up reaching the effector. And lastly, it limits the number of impulses, and that's important to prevent an overreaction to a stimulus. So how you can then speed up these impulses, one way is the Schwann cells, which we briefly talked about. Schwann cells wrap around the axon to form the myelin sheath, which does not allow the charged ions, those sodium ions and potassium ions, to pass through, going in and out of the axon. There are those gaps, which are known as the node of Ranvier, though, and that results in the saltatory conduction, which is where that action potential jumps from node to node. And that means the action potential travels faster along the axon because you're not generating an action potential at every single position along the axon. So when that impulse does reach the end of a neuron, it reaches a gap, which is known as a synapse. So the synapses are these gaps between the end of an axon of one neuron and the dendrite of another one. And at that point, the action potential is transmitted as neurotransmitters, which are chemicals, which diffuse across the synapse. So let's go through how that happens. When that action potential reaches the end of the neuron, that is known as the synaptic knob, the end of the neuron. And depolarization of the synaptic knob causes calcium ion channels embedded within the membrane to open, and therefore calcium ions diffuse into the synaptic knob. Those calcium ions cause vesicles within the synaptic knob, which contain the chemicals, the neurotransmitter, to move towards the cell surface membrane. They fuse with that presynaptic membrane, and when they fuse, that causes the release of the neurotransmitter by exocytosis into the synaptic cleft, which is what we call this gap between the two neurons. Because that neurotransmitter is only being released from the presynaptic neuron, we have a concentration gradient. So that means the neurotransmitter chemicals can diffuse down their concentration gradient, which causes them to diffuse across the synaptic cleft to the postsynaptic neuron. And it goes to the membrane of that postsynaptic neuron, which has receptors embedded within that neuron membrane. And those receptors are complementary in shape to these neurotransmitter chemicals. And as a result, the neurotransmitters bind to those receptors. Once they bind to those receptors on the postsynaptic membrane, that causes sodium ion channels on that postsynaptic membrane to open. And once those sodium ion channels open, sodium ions can diffuse into the postsynaptic neuron, causing the postsynaptic neuron to depolarize. And that depolarization can then result in the generation of an action potential, and that has then triggered an action potential in the next neuron. Those neurotransmitters do not remain permanently bound to the receptors though, because if they did, that would result in the action potential constantly being generated in the postsynaptic neuron, even if there was no longer a stimulus there. So that means the neurotransmitter has to be released from the receptor. And the way that that happens is the neurotransmitter chemicals are broken down or degraded, and that causes them to be released from the receptor. That causes the sodium ion channels to close, and that is how you then don't get any more action potentials being generated. And that broken down or degraded neurotransmitter can be recycled and returned back to the presynaptic neuron to then regenerate those neurotransmitters to be reused again. So this response is unidirectional. It's unidirectional for a few reasons. First of all, you only have the vesicle containing the neurotransmitter in the presynaptic neuron. You only have the receptors that the neurotransmitter binds to on the postsynaptic neuron, and the neurotransmitter diffuses across because of the concentration gradient. Now, an exact example of this synapse response or transmission across the synapse is a cholinergic synapse. And it's called that because the neurotransmitter is acetylcholine. So you would go through that exact same process that we described, but wherever we said neurotransmitter, you'd say acetylcholine instead. And the acetylcholine is broken down or degraded after it's attached to the receptors by the enzyme acetylcholine esterase, and that breaks down the acetylcholine into choline and acetate, which then gets recycled. Now, thinking back to when we talked about the generation of an action potential, we said that you only get an action potential if depolarization reaches at least minus 55 millivolts, that threshold potential. So when you're at a synapse, summation has to occur to reach that threshold. And summation is the rapid buildup of the neurotransmitter in the synapse so that you have enough neurotransmitter binding to enough receptors to open enough sodium ion channels to result in sodium ions diffusing in to reach that minus 55 millivolt threshold. And there's two ways that summation can occur: spatial and temporal. Spatial summation is when you have multiple presynaptic neurons all converging at one postsynaptic neuron. And collectively, those presynaptic neurons should release enough neurotransmitter to help trigger an action potential. Temporal summation is when there is only one neuron, so one presynaptic neuron converging to one postsynaptic neuron, but that one presynaptic neuron will release neurotransmitter repeatedly over a short period of time to add up to enough to exceed that threshold value. Now, at the end of your response, you have an effector, which can be a muscle cell or a gland, and that means you'd have your motor neuron reaching a muscle fibre. And the gap between the motor neuron and the muscle fibre is called a neuromuscular junction, because you've got a neuron reaching muscle. So these are specialised synapses, which we said are formed between the motor neuron and the muscular fibre. And when an action potential reaches the end of the motor neuron, it triggers the release of neurotransmitter acetylcholine into the synaptic cleft, just how we saw that happens in a typical synapse. That acetylcholine then binds to receptors, but this time the receptors are on the muscle fibre, not on a postsynaptic neuron's membrane. And that then causes sodium ion channels to open, sodium ions diffuse in, and it causes depolarization of the muscle cell membrane. This depolarization generates an action potential that spreads across the muscle fibre membrane and into tubes known as T-tubules, and it gets into the T-tubule system. So you could be asked to compare similarities between a neuromuscular junction and a cholinergic synapse. So here is our comparison. They both involve unidirectional movement. A neuromuscular junction is always excitatory, meaning it will always result in a response, whereas cholinergic could result in a response, or you do get inhibitory synapses where chloride ions are released instead. We've got the difference between where they connect. The neuromuscular junction is at the end of that response arc, whereas a cholinergic synapse can result in a new action potential generated in the next neuron. And then lastly, the acetylcholine binds to receptors on muscle fiber membranes for a neuromuscular junction, whereas the acetylcholine binds to receptors on postsynaptic membranes for a neuron. So at that neuromuscular junction, we said that the action potential causes depolarization that reaches the T-tubule system. So T-tubules are these invaginations of the sarcolemma, which is the muscle cell membrane, that penetrate deep into the muscle fiber. And they ensure the rapid transmission of the electrical signal throughout the muscle fiber, enabling uniform initiation of muscle contraction. As the action potential travels along the sarcolemma, which we can see up here, and into these T-tubules, which are shown there, it simultaneously activates the entire muscle fiber. The sarcoplasmic reticulum, or SR, is a specialized form of endoplasmic reticulum found in the muscle cells, and it is crucial in regulating calcium ion channels. And those sarcoplasmic reticulums surrounds the myofibrils, which are the contractile units, which we're going to have a look at, which store calcium ions. So when the action potential travels along the T-tubules, which are here, it triggers the release of calcium ions from the sarcoplasmic reticulum into the cytoplasm of the muscle fibers, and this increases the calcium ion concentration, which leads to the exposure of binding sites on the actin filaments, which ultimately results in a muscle contraction. So that leads us on to what muscles, or the structure of muscles, and how we get that contraction. So muscles always act in antagonistic pairs, which means there's one contracts, the other relaxes, and that happens against the skeleton to result in movement of your bones. Myofibrils are made up of fused cells that share nuclei and cytoplasm, which is known as sarcoplasm. And there is also a really high number of mitochondria because for a muscle to contract, you need ATP. So here is a myofibril, and the myofibrils are made up of sections known as a sarcomere. Muscle fibers are made up of millions of myofibrils, which collectively bring about the force to cause the muscle to contract. And myofibrils are made up of two key proteins. We have myosin and actin, and those form the sarcomere. And that's what we're going to have a look at in more detail. So this is just showing you that sarcomere. If I go back to here where we can see this whole, or a section of a myofibril, and we've got our sarcomere indicated at this point with a thin filament and a thick filament. The thick filament is the myosin, which is one of the proteins. The thin filament is the actin, which is the other protein. And this time it's shown here in blue. That is your myosin, the thicker filament. And in red here we have the actin, which is the thin filament. And it's just named that because they are thicker and thinner proteins. So we can see here in the sarcomere, we have the actin on both sides, and that is interleaved between the myosin, which is in this fixed position. So the myosin stays in that fixed position, but when a muscle contracts, the two sides here with the actin slide closer together and they slide over the myosin. And we can describe this contraction based on these different lines and zones in the sarcomere. So the A band, that is the section of the sarcomere where you have the myosin. And because the myosin doesn't change position, the A band will always stay exactly the same width. The Z lines, which we can see here, those indicate the parameters or the border of your sarcomere. And when a muscle contracts, the Z lines get closer together. So we can see that Z line's moved in here and it's moved in there. So those Z lines get closer together. That's why when muscles contract, they actually get shorter and fatter because the Z lines are moving closer together. We then have this I band. The I band is where you just have actin by itself, not overlapped with any myosin. And when a muscle contracts, that I band gets shorter because the actin is sliding over the myosin, so you have more section where it's an overlap. The H zone is where you just have myosin by itself with no actin in between it. And when the muscles contract, because the actin is sliding in between, you have a shorter H zone. And those are your key sections that you need to know. So you need to be able to label those bands and zones, and you need to know what happens to them during muscle contraction. And we're going to go through how the actin slides closer together and over that myosin in this muscle contraction. And this is what the sliding filament theory is. When that action potential reaches the muscle, we already talked about how it generates that depolarization traveling through those T tubules, and it stimulates a response because it causes calcium ions to enter, and those calcium ions can cause tropomyosin, which is shown here in orange as protein, to move to a different position. And that position that it moves to results in the actin exposing the myosin binding sites. So that thin filament actin has binding sites on it that myosin heads, which are these structures branching off the myosin, they can bind to. So once that those calcium ions have been released and the tropomyosin has been pulled out the way, the binding sites, the myosin binding sites are revealed, and the myosin heads, because they're complementary in shape, those attach. And whilst the ADP and PI are attached to that myosin head, they are the correct shape, that is the myosin head, to be able to attach to those myosin binding sites on actin, and it forms what we call a cross bridge. So we've now created a bridge between the myosin and the actin. This angle that is created in the cross bridge creates tension on the myosin head. And as a result, that tension results in this power stroke, which is when the myosin head flicks. It really slides that actin along the myosin. And in doing so, that power stroke action causes the release of ADP and PI. An ATP molecule can then bind to the myosin head, and at that point it causes the myosin head to slightly change shape so that it's now no longer complementary to the myosin binding sites, and as a result it detaches from the myosin binding sites on actin. Within the sarcoplasm there is an enzyme ATPase, which is then activated by the calcium ions to hydrolyze that ATP on the myosin head into ADP and PI, and that releases enough energy to cause the myosin head to return back to its original position, which is known as recocking. So cocking of the myosin head is the movement of it back to its original position. And as long as the calcium ions are still present, holding that tropomyosin out the way, that whole process can continuously repeat, and that will slide the actin closer and closer together until you have a full muscle contraction. So the final part of this topic is looking at the control and coordination in plants. And plants can't run away from animals. They don't have muscles, so they can't move to the shade or move to the light like animals have evolved to be able to do this. So instead, they have a different range of responses to defend themselves from herbivores trying to eat them and abiotic stresses. So the Venus flytrap is an example, and this shows a rapid response to stimulation, particularly when prey, such as insects, come into contact with its modified leaves, known as lobes. And these lobes are lined with tiny sensitive trigger hairs, which you actually can't see in this diagram, but on the red section here, that is where you'd have those trigger hairs. And when an insect brushes against these trigger hairs, it causes the hairs to bend, generating an action potential within the plant. The action potential is similar to the nervous impulses found in animals, and they travel rapidly throughout the lobes. The action potential spreads through specialized cells within the lobes called motor cells, and these cells act as a bridge between the trigger hairs and the closure mechanism of the trap. And as the electrical signal reaches the motor cells, they rapidly transport ions, such as calcium ions, across the membranes, creating a change in turgor pressure of the cells. And that change in turgor pressure within the motor cells causes a rapid change in the curvature of these lobes. And that is why the lobes end up snapping shut, and as a result, it traps the prey inside. Once trapped in that closed section, the plant secretes digestive enzymes to break down the captured prey, and that extracts nutrients from the insects, such as nitrogen and phosphorus, from their bodies. And that provides the plants with essential nutrients, which the environment may not contain because there might be nutrient-poor soil. So other responses are tropisms, and this is the term given to when plants respond via growth to a stimulus. You can have positive or negative tropisms. Positive means it's growing towards a stimulus, negative means it's growing away from the stimulus. And plants typically respond to these three stimuli: light, gravity, and water. Tropisms are controlled by specific growth factors, and one key example is IAA. And that's a type of auxin that can control cell elongation in the shoots of the plants and actually increases cell elongation in the shoot and inhibits the cell elongation in the roots. So if we have a look then at phototropisms, which is an example where it's responding to light, in the shoot, light is needed for the light-dependent reactions in photosynthesis and the leaves, which are going to be branching off the shoots. So it's an advantage, an adaptation, that the plants, the shoot part of the plant, will bend towards the light source, a positive phototropism. And the way this happens is IAA is produced in the shoot tip. The IAA is able to diffuse to other cells where it isn't produced and cause cell elongation. And if there's unilateral light, the IAA will diffuse towards the shaded side, because unilateral means the light source is only on one side, which is what we can see here in image 2. The light source is slightly over to the right. So all of that IAA is going to diffuse to the shaded side, which is on the left. That will mean we'll get more cell elongation on the left-hand side compared to the right. So the left side of the plant is going to be longer than the right-hand side. And as a result, the plant starts to bend towards that light source, which is useful because the plant will then get more light energy, so it can absorb more of that light energy for the light-dependent reactions in photosynthesis. Now, in the roots, we have the opposite effect. Roots do not photosynthesize, so they don't require light, but instead it's an advantage if the roots are deeper in the soil so they can anchor in, and also it helps to reach more water. So in roots, if there's a high concentration of IAA, it inhibits cell elongation, and that causes the root cells to elongate more on the lighter side, and so the root ends up bending away from the light source, which is what we can see here in this animation, and that is negative phototropisms. Gravitropism, this will then occur in the roots and shoots differently. In the shoots, we can see that the IAA diffuses from the upper side to the lower side, so more of it's now on the lower side, so it goes down with gravity. That means that we get more cell elongation on the lower side, and that causes that side to be longer, and therefore it bends upwards. And in this case, it's a negative gravitropism because the plant will be growing against gravity. In the roots, IAA moves to the lower side of the root, so that the upper side elongates, and the root bends down towards gravity and anchors the plant in. And this is a positive gravitropism because the root is bending towards the stimulus, which is gravity. So plant hormones then control a range of responses in plants, such as the ripening of fruit, germination of seeds, and as we just saw with the IAA, the lengthening of stems and when also leaves drop. Examples of plant hormones include auxins, which is what we just went through with IAA, ethene, which is a gas that causes fruit to ripen, and gibberellin, which stimulates seed germination, stem elongation, and pollen tube growth in fertilization. So a bit on gibberellins then. Gibberellin is needed for germination of barley seeds, as an example, by initiating and coordinating the physiological processes for growth. When a barley seed absorbs water, it triggers the synthesis and release of that plant hormone gibberellin. And these gibberellins act as signalling molecules within the seed, resulting in a series of events that leads to germination. One of the primary functions of gibberellin during germination is to stimulate the production and activation of enzymes involved in breaking down stored food reserves in the seed. And barley seeds store energy in the form of starches and proteins. Gibberellins prompt the release of enzymes such as amylases and proteases, which will then hydrolyze those stored reserves into smaller, more accessible molecules. And this breakdown by enzymes provides the embryo plant with necessary nutrients and energy sources, such as glucose and amino acids, to fuel the metabolism during germination. Additionally, gibberellins promote cell elongation and expansion, which are essential processes for the emergence of that embryonic shoot, which we can see in the image here. And by stimulating cell elongation, gibberellins enable the embryo to push through the seed coat and emerge to the surrounding soil. And this growth is crucial for establishing young plants and enabling it to access light and nutrients for further development through photosynthesis. Gibberellins also play a role in overcoming seed dormancy, which is a state of suspended growth and metabolic activity that prevents germination if there are unfavourable environmental conditions. So gibberellins counteract the inhibitory effects of other hormones such as ABA, which maintains seed dormancy. And by promoting germination, gibberellins help the seed to respond appropriately to favourable environmental cues such as moisture and temperature, and therefore initiate the growth process. Auxins play a key role in cell elongation, like we saw, and this is by stimulating proton pumping to acidify cell walls. And this process, known as acid growth, is essential for cell elongation and expansion in various plant tissues such as the stems, roots and young leaves. So the auxin promotes the activity of proton pumps, particularly H+ in the ATPases, and that is located in the plasma membrane of plant cells. These proton pumps actively transport hydrogen ions, which are protons, from the cytoplasm into the cell wall space or apoplast. And as protons are pumped into the cell wall space, they accumulate, leading to a decrease in pH, which is the acidification of the cell wall. This acidification softens the cell wall by disrupting the bonds between cellulose microfibrils, hemicelluloses, and pectins, which are the main components of the cell wall matrix. That acidification of the cell wall increases its plasticity, making it more flexible and susceptible to deformation. This softening allows the cell to expand more easily under turgor pressure, generating the influx of water into the cells, and as a result, the cell elongates. And that's how we get that cell elongation. Topic 16, which is looking at the passage of information from parents to offspring, or in other words, inheritance. And we're going to start by looking at these key terms for inheritance. So haploid, also represented by the letter N, means that you have one set of chromosomes in those cells. Diploid cells, or 2N, means you have two sets of chromosomes. And homologous pairs of chromosomes are two chromosomes that contain the same genes in the same position. Now we're going to look at meiosis first of all. And meiosis is a type of cell division. It's known as reduction division because it results in the production of gametes, meaning you end up with half of the number of chromosomes compared to the parental cell. This is crucial because when two gametes fuse together during fertilization, the resulting zygote will then have the correct diploid chromosome number. And without that reduction division, the chromosome number would double every single generation when fertilization happened, leading to an exponential increase in chromosomes and the number of genetic material. Now, unlike in mitosis, there are two nuclear divisions. That's why we end up with reduction in the chromosome number. And we get four genetically different haploid cells. So how that happens then, as we said, we've got two nuclear divisions. We get four genetically different haploid cells. We have two rounds of division that are referred to meiosis 1 for the first round and meiosis 2 for the second round. Both stages include the same stages: prophase, metaphase, anaphase, telophase, and then cytokinesis to split the cytoplasm and the cell membrane to create two new cells. And here's just a reminder of these two key terms, haploid and diploid, because that's going to come up as we go through the stages of meiosis. Now, the key stages that you need to know about actually happen in Meiosis 1. So the way we get these genetically different haploid cells, the genetic difference is introduced by independent assortment of homologous chromosomes and crossing over, both of which happen in Meiosis 1. So crossing over, this happens in Meiosis 1 when the homologous chromosomes pair up at the equator. And when they pair up at the equator opposite each other, we call that a bivalent. So this here would be a bivalent because it's two homologous chromosomes aligned next to each other. What can happen, not all the time, but sometimes when that bivalent forms, the non-sister chromatids, which means the chromatid from the two different chromosomes, can cross over when they're in that bivalent. And the exact position where they cross over is known as a chiasma. Where those non-sister chromatids cross over, it can put tension on that chromatid, causing parts of the chromatids to break and then swap, and they're exchanged. And because they've now swapped a section of that chromatid, they have exactly the same genes because they're homologous chromosomes, but they might have had different versions or different alleles of those genes. And that results then in new combinations of alleles in the resulting gametes that will be made from these chromosomes. Independent assortment also happens in Meiosis 1, and it's in metaphase 1 when the homologous pairs are lining up at the equator to make those bivalents. But this time the concept is it's random which side of the equator the homologous pairs align. So whether you have the maternal or the paternal on the left or the right for all of those homologous pairs. So we can see here we've got one option where you've got all of the blue on one side, all of the red, but we've got a different combination in this option and a different combination here and a different combination there. And because it's random which side of the equator they go, you're going to end up with different combinations of maternal and paternal chromosomes in the gamete, which introduces genetic variation. And you can actually calculate the number of possible combinations you could get of how those homologous pairs could align, and therefore how many different possible gametes you could create by using the formula 2 to the power of n, where n is the number of homologous pairs. And it's 2 because we have 2 chromosomes in that pair. So for humans, we have 23 homologous pairs. So to work out how many possible different gametes we could create because of independent assortment of homologous chromosomes, it'd be 2 to the power of 23, which gives us over 8 million different combinations. Next then, we go on to the roles of genes in determining the phenotype. Here's a selection of essential key terms you need to know for inheritance. So we've got genotype and phenotype, the genetic constitution of an organism, so it's the alleles it has for a gene, whereas the phenotype is the expression of the genes and its interaction with the environment. Homozygous means a pair of homologous chromosomes carrying the same alleles for a single gene, whereas heterozygous is when you have 2 different alleles for a single gene. We've got our recessive, dominant and co-dominant alleles. Recessive alleles are only expressed if there are no dominant ones present. Dominant alleles are always expressed. And co-dominance is when you have 2 alleles that are equally dominant, and therefore if you have a copy of both in your genotype, both will be expressed in the phenotype. Multiple alleles is when there is more than 2 possible alleles for a particular gene. Sex linkage is when a gene is located on the X chromosome. Autosomal linkage is not about the sex chromosomes, but it's when genes are located on the same chromosome, but not a sex chromosome. Epistasis is when one gene modifies or masks the expression of another gene. Monohybrid inheritance is when we look at the inheritance of one particular gene, and dihybrid inheritance is when we look at genetic crosses for 2 particular genes. So these would be good to turn into flashcards to test yourself and to learn. Now, you commonly have to do genetic diagrams to determine the probability of different either genotypes or phenotypes in the offspring based on parental genotypes or phenotypes you're given. And we use different coding to represent the alleles. So this is just a summary of the accepted coding. In monohybrid inheritance, which is what you would have done at GCSE, when you're looking at the inheritance of either a dominant or recessive allele for one particular gene, we pick one letter, and that represents the gene, and you have the capital letter, which represents the dominant allele, lowercase letter represents the recessive allele. In codominance, because both alleles are dominant, it wouldn't work to use one letter that were capital or lowercase, because they'd both have to be capital and therefore it would look the same. So that's why we changed the coding system, and we use a base letter that represents the gene, and then we do a superscript letter which represents the allele. And we use two different letters, both capitals, to represent the two different codominant alleles. We have to do the same thing in multiple alleles because you have more than two alleles for a gene. Therefore, we can't just do capital and lowercase with the same letter. So that's why here we can see we've got the base letter for the gene and we've got the three possible alleles for blood groups shown there. Sex linkage, we do the base letter showing the chromosome and then the allele goes superscript. And you don't get the sex-linked genes on the Y chromosome, so you never have an allele written next to the Y, you only have an allele written next to the X. Autosomal linkage, this is just using the same system as the monohybrid, but that will always be a dihybrid example, so that's the inheritance of two genes, that's why we've got two different letters, but it should be capital or lowercase for dominant and recessive. Same with epistasis, that's always a dihybrid example because one gene can mask the expression of the other gene. But you'd pick one letter for one of the genes, another letter for the other, capital and lowercase to represent dominance and recessive. So let's go through some genetic cross examples to look at how this all works. Starting with monohybrid. This one we've got cystic fibrosis is caused by a recessive allele. And we've got if two carriers reproduce, what is the probability that they'll have a child with cystic fibrosis? So here we have the parental genotypes. They're both carriers, so they need a capital and a lowercase, a dominant and a recessive. I've then done my Punnett square, showing the possible gametes for both parents, join those gametes together to show the four possible genotypes of the offspring. And then I've put in what the genotype for someone who is a cystic fibrosis sufferer must be if it is caused by a recessive allele. So from that, we can see we have 25% probability of a child having cystic fibrosis. If they then asked what is the probability of it being a girl, which they did here, we then need to times by a half because it's always 50% probability of having a boy, 50% probability of having a girl, or male or female. Then if we have a look at this codominance example, cows can be red, white or roan. We call this colour red when we're describing cows. Red and white are both dominant, and you only get roan, which is this mixture of white and red, if an individual has the codominant genotype. So they have a red and a white allele, so both are expressed in the phenotype. And in this example, we're told that we have two roan cows reproducing, and we're asked what is the probability they'll produce red offspring. So here are the parents' genotypes. If they're roan, they must have one of each allele, a red one and a white one. We've then got our Punnett square showing the parental gametes on the outside, just as a single letter, because you only have one allele for each gene in a gamete. Joining together those gametes to show the four possible genotypes, and I've written in the box the four matching phenotypes. And from that we can see we have 25% probability of having a red cow with those parents. Blood groups is an example of codominance and multiple alleles. There are three alleles, blood group, the allele for blood group A, an allele for blood group B, an allele for blood group O. But A and B are both dominant. So if an individual has A and B, they'll have the fourth phenotype, which is blood group AB. Blood group O, or the allele that codes for blood group O, is a recessive allele. So you're only blood group O if you have two copies of that recessive allele. And that's why you could have allele B and allele O and still be blood group O, because B is dominant over O, And the same with blood group A. Allele IA is dominant over allele IO. So here's our potential genotypes. There's our potential phenotypes. And in this example, we've got parents with blood group AB and blood group O reproduce. IA and IB are co-dominant and IO is recessive. What's the probability they'll have an offspring with blood group A? So first of all, we need to work out the parents' genotypes. And if they're blood group AB, one parent must be IA-IB, and blood group O, one parent must be IO-IO. So then if we split it up to see the gametes that both parents would be able to produce, we can then do our Punnett square to see the 4 possible genotypes. And then in the box, I've also written the corresponding phenotype. And from that, we can see we've got 50% probability of blood group A. We're now moving on to sex linkage. We're going to look at the example of colour blindness. Colour blindness is caused by a recessive allele found only on the X chromosome. And we've got the example of if a non-colourblind male reproduces with a female carrier of the allele, what's the probability of their children being colourblind? So the female is a carrier, which means they must have 2 different alleles, so the capital and the lowercase. And recessive is colour blindness, so that is our colour blindness allele. And they're reproducing with a male, so they are XY, and the male does not have colour blindness. So the males only have one version of the gene, and the version they have is the capital R for non-colourblind. So if we have a look at the Punnett square, we can see the gametes on the outside, the 4 possible genotypes, and the matching phenotypes. So this would be a biological female with colour vision, because it's a dominant allele, biological female with colour vision, and that'd be a colourblind biological male, missed off the biological male there, and then the biological male with colour vision. So the probability of having a child being colourblind is 25%. Next is epistasis, and that was the definition we said is where one gene influences the expression of another gene. An example of this is coat colour in mice or coat colour in Labradors. Fruit colour in vegetables is another one. So colour is quite a common example. So if we go through the coat colour in Labradors example, one of the genes controls whether pigment will be expressed, so produced. The dominant allele for that gene means the pigment will be made. The recessive allele codes for no production of pigments, and in which case for a Labrador, if they have 2 copies of this recessive allele, it will mask the expression of the second allele, and they will always be yellow in colour, or golden, whatever you want to call it. Gene 2 is the gene responsible for what colour the pigment will be. So capital B would be a dominant allele, and that one codes for black fur. Recessive allele is for brown fur. So here's our 3 possible phenotypes, but gene 1 can mask the expression of gene 2. Because if an individual is lowercase e, meaning 2 recessive alleles, it doesn't matter what combination of alleles they have for gene 2 is not going to be expressed. So if we go through an example, here we have 2 parents heterozygous for both genes. And because they've got at least one capital E, that means the pigment will be expressed. And because they've got a capital B, that means the pigment is black. So this would be 2 black Labradors. The possible gametes that they could both produce would be all of the options of the capital E, lowercase e, capital B, lowercase b in your gametes, because you only have one copy of each gene in each gamete. So here's our 4 possible gametes for both parents. That then gives us this Punnett square. It's a large one because there's 4 gametes for all of them. And we can then put in what the possible genotypes would be and the possible phenotypes. So we can see here any that have a capital E are going to be either black or brown. Any that have 2 recessive E alleles will always be yellow, and it masks the expression of that second gene. So in this instance, we've got a 9:4:3 ratio of those phenotypes. So a dihybrid cross is where the inheritance of two genes is considered at the same time, which we actually just saw there in that epistasis example. A common example linked to this is Mendel and his peas. So he looked at pea plants, considering the gene for colour and the gene for whether it was round or wrinkled. Only he didn't know genes at that point. He was just talking or noting down the colour and texture. And when we do genetic crosses, we should always show and label where possible the parental phenotype, parental genotype, possible gametes, the offspring genotype, correctly match up the phenotype to those genotypes and give the proportion of each phenotype. And that's why often in the exam they give you this space to fill in each of those criteria. So we've got a round yellow and a wrinkled green. And in this example, we are being told that the round yellow is homozygous dominant for both of those genes. Wrinkled green would have to be homozygous recessive for both because wrinkled and green are recessive. Now, you aren't expected to know that. That's information you would have had to have been told. Then we work out the possible gametes. And in a dihybrid example, every gamete should have one copy of each letter. So one of the R's, one of the Y's, to indicate one copy of each gene. Now for this parent, they can only produce gametes with a capital R, capital Y, which is the two dominant alleles. And this parent can only produce gametes with the two recessive, so recessive R, recessive Y. We can then do a Punnett square, but actually for this one, we don't need to do a Punnett square because if this parent can only donate this gamete, this parent can only donate that one. If we join them together, every single option on the Punnett square would come out with this heterozygous offspring genotype. So 100% would have that genotype, which would mean 100% would be yellow and round, but heterozygous. So you can actually cross those together now, and this commonly is shown. If you were then to cross F1, which is the first generation, which is what we just created together, we've now got two parents who are both heterozygous for both genes. For this, we now have these four possible gametes for both parents, and now we're going to end up with one of these large Punnett squares again, where we'd have to put in all of the possible genotypes, combining those gametes, and then we can put in all of those possible corresponding phenotypes, and then we can work out the ratio or proportion of the phenotypes. And we have 9 yellow and round, 3 green and round, 3 yellow and wrinkled, and 1 green and wrinkled. So a 9-3-3-1 ratio. And in any dihybrid cross, if you are given a scenario where both parents are heterozygous for both genes, you will always get a 9-3-3-1 ratio. Unless there is autosomal linkage, which we're going to have a look at, or if there's crossing over in meiosis, or if epistasis has occurred as well. So crossing over in meiosis, that creates new combinations of alleles in a gamete. So that means what you predicted in your Punnett square may not happen. And this links back to what we said right at the start of the video. Crossing over introduces new possible combinations of alleles in the gametes, and therefore we don't get what we expected from our Punnett square. I'm going to come back to that concept once we've gone through autosomal linkage. And we said autosomal linkage is when two different genes are on the same non-sex chromosome. So we can see here a homologous pair of chromosomes, gene 1 and gene 2 are on the same chromosome. So they are linked autosomally. We're going to apply the example we've just done. So gene 1 is for whether it's yellow or green. Gene 2 is for whether it's wrinkled or smooth. And we said that the parents were both heterozygous for both genes, which we can see. This is the homologous pair of chromosomes for one of the parents, and they've got the capital on the recessive, the capital on the recessive. It just so happens to be, because they're linked, the two dominants, so the two capitals, are on this chromosome of the homologous pair, and the two recessive alleles are on the other chromosome in the homologous pair. And that means we can't actually make 4 possible gametes now. We can only make 2 possible gametes, because in meiosis, when these homologous pairs are separated, that will mean this will go on to make 1 gamete, which will have to have the capital R and the capital Y, and this chromosome will go on to make another gamete, which means that one will always have lowercase r and lowercase y. And therefore, these are the only two gametes that this parent can make, rather than the four possible ones that we looked at initially. So here were the four possible ones that we said initially if there was no autosomal linkage. But because there is autosomal linkage, these are the only gametes that the parent can produce. And if we have two parents who are the same, reproducing with those two possible gametes, these are the four possible genotypes we get and the corresponding phenotypes. So our ratio is now 3 yellow and round to 1 green and wrinkled. So with two heterozygous parents, you would expect to get a 9:3 to 3:1 ratio, unless there's autosomal linkage, when you could get a different ratio. And for this particular example, we got a 3:1 ratio. Now, we did also say unless there's crossing over that happens. So sometimes you might get an autosomal linkage exam question where you're led through all the information we've just gone through to work out you'd expect a 3:1 ratio. But they then might say, however, scientists did this cross, and what they actually observed was these phenotypes in this proportion. And you could be asked to explain their results. And it's only possible to get those two extra phenotypes if crossing over occurred in meiosis between these homologous pairs to create new combinations of gametes. So here we can see those linked genes. These were the two recessive ones on one of the homologous pairs. Here were the two dominant ones on one of the homologous pairs. They now look like double structures because this is showing after DNA replication. And in meiosis 1, when they line up at the equator to form a bivalent, we can see the non-sister chromatids crossing over to form a chiasma, break and swap, and that then means when they do then separate in meiosis 1 and meiosis 2, ultimately we get 4 gametes which have new combinations of alleles in the gametes. So to explain these results then, we'd have to say yellow and round and wrinkled and green are the most commonly observed because those are the genotypes that are a result of fusing the capital R and Y and the lowercase r and Y together. And those were the ones that are most common because they were not formed by the crossing over, and crossing over is actually quite rare. Now, out of the yellow round and green wrinkled, we've got fewer green and wrinkled because that's the recessive combination. So you'd have to have 2 copies of the R's, 2 copies of the lowercase y's to be green and wrinkled. So that's why there's fewer green and wrinkled compared to yellow and rounds. Then why we don't have very many at all of the middle two, those two are a result of crossing over to create the capital R lowercase y and the lowercase r capital Y gametes. And crossing over doesn't happen all the time, it's rare. And therefore we wouldn't get very many of these two genotypes, which is how we'd get those two phenotypes. So that would be your explanation of those results, combining the concept of autosomal linkage and crossing over. Now, throughout that, I was talking about what we expected to see versus what we actually observed, and that leads us into using the statistic of chi-squared in inheritance. So chi-squared is the statistic to investigate differences between frequencies, and it can be used to determine is there a significant difference between the frequency you expect and the frequency you observe. And in inheritance, that links to what we expect from the Punnett square, and we can compare that to what we actually observe if you count literally the phenotypes that are there. And here's an example of this. Corn can be purple or yellow or smooth and wrinkled in terms of colour and texture. Using the results of the Punnett square and then counting the number of actual corn kernels of particular colours and textures, you can look at whether the frequency you expected is significantly different to the frequency you observed. So we're told here that purple is dominant, yellow is recessive, 2 heterozygous parents were crossed. This is just dihybrid, simple genetics. There's no autosomal linkage. And the expected ratio is 3 purple to 1 yellow. So that is our expected ratio. And we're told that a student then counted them and they had 21 purple kernels and 13 yellow kernels. Does this follow the expected ratio? So what we would then need to do is calculate the chi-squared, but I'm actually jumping straight to the result here so you can see how we'd use this as a conclusion. So our null hypothesis would be, for this example, there's no significant difference between the expected and observed frequency of the colour of the corn kernels. The chi-squared value was then calculated to be this. We've got 1 degree of freedom because there were 2 categories. So if we read across to 1 degree of freedom at p equals 0.05, that means our chi-squared value is lower than the critical value. And therefore we have more than 5% probability that the difference between the observed and the expected is due to chance, and therefore we have to accept the null hypothesis, meaning there is no significant difference between what we observed and what we expected. So in other words, the frequency of the corn kernels that we observed does match what we expected from our Punnett square, and it confirms that Mendelian genetics ratio of 3:1. Test cross is the final thing we're going to look at, and these are used to determine the genotype of an individual expressing a dominant phenotype by crossing it with a homozygous recessive individual. And this helps in revealing whether the dominant phenotype an individual has is determined by a genotype that's heterozygous or homozygous dominant. Because if you have the dominant phenotype, that could be someone who's homozygous dominant or heterozygous. So if we use an example of Mendelian genetics or inheritance, specifically the inheritance of flower colours in pea plants, where purple is dominant and white is recessive, we could perform a test cross to determine the genotype of a purple flower. So what we would need to do is cross the purple flower plant that we're trying to identify the genotype of with a white plant, because we know that white plant has to be homozygous recessive. From the results of the Punnett square or the offspring breeding together, we can then work out whether the parent was homozygous dominant or whether they were heterozygous. Because in this Punnett square we've crossed the purple flower with a white flower, and because we do have a white flower here, that would only be possible if the purple parent was heterozygous, because to get a white flower, both parents have to have a white allele. So in these test crosses, if you get any offspring that have the recessive phenotype, it confirms the parent that was the dominant trait must be heterozygous. And that's what we've just got here explained. So you can pause, read through that bit, but it's what I was just talking through on that last slide. Topic 17 then, we're going to be looking at selection and evolution, starting with variation. So the genotype of an organism is its genetic constitution, which basically means the alleles that they have for a particular gene. Whereas the phenotype of an organism is the observable characteristics determined by both the genotype and environmental contributions as well. So for example, you have a combination of genes that genetically determines the maximum height you could grow to. But the environmental factors, such as your diet, will determine whether you reach that maximum height. And that would be your phenotype, which would be your height. An example for plants is chlorosis, so the yellowing of leaves. Plants have genes for chlorophyll production. However, environmental factors, such as lack of light or mineral deficiencies and viral infections, can reduce the production of chlorophyll. So thinking about this concept then of variation, we can categorize it as continuous and discontinuous variation. And data representing continuous variation would be presented as a histogram, whereas data representing discontinuous data would be represented as a bar chart. And the differences between these two types of variation are summarized in the table below. So continuous variation are characteristics with continuous values. So for example, your height or the height of a plant or birth weight. Discontinuous variation would be categorical values, so discrete values. So for example, which blood group you would have. Continuous variation is caused by your genetics and interactions with the environment, whereas discontinuous is just genetics. Like your blood group isn't going to change because of what environment you live in. And then the final comparison is the number of genes involved. Continuous variation, the reason that there are a continuous range of traits that people could have for that particular characteristic is usually because there are many genes and many alleles for those genes involved, so polygenic. Whereas discontinuous variation is normally controlled by one, possibly two genes. So then we go on to looking at natural and artificial selection. Natural selection is the process that leads to evolution in populations. And evolution is the change in allele frequency over many generations in a population. Natural selection results in a species as a whole becoming better adapted to their environment, and those adaptations could be anatomical, physiological, or behavioural. So let's just go through then the process of natural selection. First of all, you always have variation, genetic variation within the population. And that is because new alleles for a gene are created by random mutations occurring. If this new allele provides a selective advantage, meaning it increases the chances of the individuals who have that mutated version of the allele to survive in that particular environment, then they're more likely to survive and reproduce, and therefore they pass on potentially that advantageous allele to the next generation. As a result, if this continues over many generations, that mutated version of the allele will now become more frequent in the gene pool of that population. And evolution is a change in allele frequency over many generations. So that is how natural selection results in evolution. Now, there are different outcomes of natural selection or types of selection, known as stabilizing, directional, and disruptive. And directional selection, this is when we can see here the y-axis would be representing the frequency of a particular trait or allele. The x-axis is that trait or alleles. And before selection, the middling trait would be the most common, and the two extreme traits are less common. But directional selection is when, in natural selection, whatever the allele that has the selective advantage, it is now one of the extremes. So in this case, it's the extreme at this end, but it could have been the one at this end. And therefore, over many generations of the individuals with the extreme trait being the ones survive, reproduce, passing on their allele, over many generations, we end up with a loss of the extreme trait at the other end and the middling trait, and far more individuals will now have that one extreme trait. And this occurs when there's a change in the environment. And an example of this is antibiotic resistance. So if we think about resistance as a scale rather than resistant or not, we've got this extreme would be not resistant, middling resistant, very resistant. The bacteria that were very resistant are more likely to survive because they're resistant to antibiotics. Stabilizing selection, this is when whatever was the modal trait, so the most common trait, that remains the trait that provides a selective advantage. So stabilizing selection occurs when there is no change in the selection pressure or the environment. And as a result, even more individuals over many generations will have that middling trait. The extreme traits are lost, so we end up with this taller peak, which is narrower because we've lost those extreme traits. And so the standard deviation decreases because we have less variation around that mean. The final type is disruptive selection. And disruptive selection is when we have both extreme traits provide the selective advantage, and therefore individuals that have either of those extreme traits are more likely to survive, reproduce, and pass on those alleles. So those extreme traits both become more common, and the middling trait is almost completely lost. And as a result, that allele frequency changes and more individuals have those extreme traits. Now, if this happens for many, many, many generations, it can ultimately lead to speciation. And what we mean by this, we'll go on to in more detail, but essentially those curves will become two separate curves, and this bit would have been lost altogether. So we now essentially have two separate gene pools, and they'd be so genetically different that they are counted as different species, and therefore it is speciation. So next, in the consideration of what genetic drift is, this is the change in the allele frequency within a population from one generation to the next. And there will always be genetic drift from one generation to the next because we do not produce cloned offspring. But it is the continual and substantial genetic drift that counts as evolution. And by that we mean there has to be a continual and substantial change in the allele frequency for it to count as evolution. And the smaller a population is, the bigger the impact allele frequency changes have proportionally. And this is why evolution occurs more rapidly in smaller populations. Next then, it's the concept of genetic bottlenecks and the founder effect. And these two processes have different causes, but the same outcome, which is a reduced gene pool. Genetic bottlenecks are caused by events that kill almost all of the population, leaving only very few individuals that survive. So we can see an example here. This might have been the original population size. An event happened, which maybe it was some kind of natural disaster that killed off almost all of the population. Now the population size is very small, so we don't have very many individuals at all. Because we don't have very many individuals, the original population gene pool might have had lots of different alleles, but by chance, the ones that survived, which we can see in this example were the green and yellows, we now have very few different alleles for a particular gene. So the gene pool has decreased. And that's what we've got written here, that many alleles for the genes are lost. And that means the remaining breeding populations are going to be reproducing and passing on the same alleles. And as a result, we get this lack in genetic diversity. And as a result, more genetic diseases will exist in that population because you have few individuals reproducing, which have very similar genetics, and therefore they'll be constantly passing on the same alleles, which if they happen to code for inherited diseases, you'll continually be passing on that genetic disease. The founder effect is when a few individuals from an existing population will migrate to an isolated area. Now, this also results in a small gene pool and a small population size reproducing together. So you get exactly the same effect. You lose some of the alleles. You constantly have the same individuals reproducing together, passing on the same alleles. So you have a lack of genetic diversity. It's just the cause is different. Instead of it being due to many individuals dying out, it's a small number migrate and isolate themselves. And that's actually what happened with the Fugates in Kentucky. They isolated themselves to a particular area in Kentucky, and some of the individuals that went had a rare genetic allele that codes for blue skin if you have two copies of that recessive allele. But because of the limited gene pool and the number breeding together was very, very small, that meant that that recessive blue allele became quite frequent in the population. And you then had many individuals in that population who had two copies of this blue skinned allele, and it resulted in a population with many of the individuals having blue skin. So thinking about allele frequencies, the Hardy-Weinberg principle is a mathematical model which can be used to predict allele frequencies within a population. So just to recap on some of these key terms, the gene pool is all the alleles of all the genes within a population at one time. Population is all the individuals of one species in one area at one time, and they can reproduce to make fertile offspring because they're the same species. And then the allele frequency is the proportion of an allele within the gene pool. So Hardy-Weinberg involves two separate equations using these different components. We've got p squared plus 2pq plus q squared equals 1. And all of those parts are the different possible genotypes. So p squared is homozygous dominant. So if you have two copies of the dominant allele, 2pq is one dominant, one recessive allele. So you're heterozygous. And homozygous recessive is q squared. And because all the individuals in the population have to have one of those genotypes, that's why if you add them all together, it equals 1. This equation down here is for all of the alleles, and you either have the dominant allele, which is represented with the letter P, or the recessive allele, which is represented with the letter Q. So all of the alleles for that gene in the population would be P plus Q, and that equals 1. So those are the two equations, but it's going to make a lot more sense if we go through an example, because you could rearrange these equations to work out the frequency of the dominant allele, the frequency of the recessive allele, or the frequency of any of those genotypes in a particular population. So let's say for this example, cystic fibrosis is caused by a recessive allele. That would be the letter Q, recessive allele. 0.02% of the population in the UK suffer from cystic fibrosis. What proportion of the UK are carriers? So always have this information at the top in mind, and always start by working out what part of the equation do they want you to determine, what part of the equation have they given you. So first of all, we've been told that cystic fibrosis is caused by a recessive allele. So if 0.02% of the population have cystic fibrosis, they must be homozygous recessive individuals, which is Q squared. So that means Q squared is 0.02%. We always convert it out of the percentage though. So that is Q squared. They want you to say what proportion of carriers. Carriers would be heterozygous, so 2pq is what we are working out. So to work out 2pq, we need to know what Q is worth and what P is worth. So Q squared is 0.002. So if we square root that, we've got what Q is. And we can then use this formula, P plus Q equals 1, to work out what P is. So P is 0.986. So we want to know what 2pq is. So all we have to do is 2 times P times Q, and that equals 0.03. So that is what proportion of the UK are carriers. Sometimes they might ask you to convert that back into a percentage, and in which case that would be 3%. So genetic variation can be as a result of natural selection or artificial selection. We've already talked about natural selection. We've said that there is always genetic variation within the population because of random mutations creating new alleles. And then because of the environmental pressures, different alleles will have the selective advantage and be passed on. And those selection pressures could be predation, disease, competition for resources. But those organisms that have phenotypes providing the selective advantage are the ones that are more likely to survive, therefore reproduce and pass on those favourable alleles to the next generation. So you get evolution occurring. Artificial selection is when humans are selecting which plants or which animals are going to breed together. So humans are selecting what they deem to be the favourable characteristics rather than it being naturally selected by the natural selection pressures. And this manipulates the gene pool so that favourable alleles become more common and less favourable alleles become less common. Now, this was particularly prevalent in creating dog breeds with particular features such as pugs, which we can see up here, where humans selectively bred dogs that would have what was deemed to be really cute features, like squashed up flat faces. But because of this artificial selection, selecting for what we thought looked cute, it's actually resulted in a lot of medical issues linked to breathing difficulties for pugs because they have such flat nose, they struggle to get in enough air for ventilation. So some other examples of artificial breeding that you need to be aware of is the introduction of disease resistance to varieties of wheat and rice plants, inbreeding and hybridization to produce vigorous, uniform varieties of maize plants, and improving the milk yield of dairy cattle. And the top two are artificial selective breeding that we were pretty much just thinking about in terms of the pug example. But then this one is slightly different because we've got inbreeding and hybridization. So for these three examples, just pause, have a read through what the steps are involved. You can always take a screenshot and print this to have in your notes. So finally, then we go on to evolution. And evolution, as proposed by Charles Darwin, can lead to the formation of a new species from one existing species over time through change in the gene pool over many generations. So genetic variation with populations within the population arises from random mutations that we've talked about. And from that, we get natural selection occurring, where we have the advantageous traits are more likely to result in the individual surviving, reproducing, and passing on. But if you have, over time, reproductive isolation, which is when you have one species, one population of that species, but they become isolated so that they are no longer reproducing together, whether they're isolated because of some geographical barrier or differences in their behaviour, you get speciation, which is the formation of a new species. And by comparing the DNA base sequences of the same gene in different species, you can examine evolutionary relationships. So the more similar the DNA base sequence, the more closely related two different species must be. And that's because they must have evolved from a common ancestor more recently, as there are fewer differences in the DNA base sequences, and therefore there must have been less time since they evolved for mutations or different mutations to accumulate in those different species to create the changes in the DNA base sequence. So speciation, then, we said it's the process of creating a new species. It occurs when one original population of the same species become reproductively isolated, whether geographically or due to their behaviour. And this isolation means there are now two populations of the same species, but they're not breeding together. And because they're not breeding together, this can result in the accumulation of differences in the gene pools, because they're going to be reproducing separately with individuals in those reproductively isolated populations, so different alleles are going to be passed on. And eventually, they could become so different in their gene pools to the extent that the two populations would be unable to interbreed to make fertile offspring. And that's when they're then classed as two different species. There are two different ways that populations become reproductively isolated, either due to a geographical barrier, which is known as allopatric speciation, or it could be changes in the reproductive mechanisms and behaviours. So even if they're in the same location, they're not reproducing together, and that is sympatric speciation. So a bit more on allopatric, where it's due to geographical isolation. We could have a population becoming geographically isolated over time, maybe by a new mountain range or new bodies of water separating land masses. This separates the original population into two, which are now unable to reproduce because of that geographical barrier. Both separate populations will continue to accumulate different beneficial mutations over time to help them survive in their environment, which are likely to vary. And due to this accumulation of DNA differences over time, the two populations become so genetically different that they'd be unable to interbreed to make fertile offspring. So they're now classed as two different species. Sympatric speciation is when they are in the same area, but they are reproductively isolated due to differences in behaviour. And this could be because of a random mutation within the population that could impact reproductive behaviour, for example, and it might mean they perform different courtship rituals, or it might be that they become fertile at different times of the year, and that's the same with plants. They might suddenly have different flowering times of the year. And due to this reproductive isolation, these individuals will not reproduce together, and there'll be no gene flow between those two groups in the population. And over time, those reproductively isolated populations will accumulate different mutations to the extent that their DNA will become so different that they cannot interbreed to make fertile offspring anymore if they were to start breeding together, and therefore they're classed as two different species. Topic 18, we're going to be looking at classification, biodiversity and conservation, and we start with classification. So there's different ways to define a species. You could have the biological species concept, which is a group of organisms that can reproduce to create fertile offspring. The morphological species concept, which is a group of organisms that may have physical features in common that are different to other species. Or the ecological species concept, which is a group of individuals of the same species living in the same area at the same time. And classification is the grouping of organisms according to their similarities. Individuals can be classified according to their physical and biochemical similarities, and they're grouped in hierarchies. And a hierarchy is when you have smaller groups, placed within larger groups, but there are no overlaps between those groups. And in classification, each hierarchy is known as a taxon, or taxa for plural. There are millions of species, and the reason we use classification is so that we have a system to organise them. And the reason for this need to organise them is to help us in identifying species, and that will help us to understand evolutionary relationships between species and to keep track of changes in population size. And it also enables scientists to share data on these different groups within different countries because the classification system is universally used. So here is the Linnaeus classification system, which is based on a hierarchy, and we have these following taxa. Domain is the broadest way to group. Then we have kingdom, phylum, class, order, family, genus, and species. And you do need to remember the name of all of these and the order. So you might want to come up with a mnemonic to help you remember them. One common one that I've heard people use is, does King Philip come over for good soup? And that would help you to remember the order that those occur in, and then you just need to remember all of those different names of the taxa. And for the 3 domains, this, as I said, was the broadest way to classify or to group. And all life forms fit into either the Archaea group, the Bacteria group, or the Eukarya group for these domains. And bacteria, this is the domain encompassing all prokaryotic organisms, except those that are categorised as Archaea. Archaea, this is the domain comprising prokaryotic organisms, but they resemble bacteria, sharing some of the characteristics with eukaryotes as well. And eukarya, that is the domain housing all eukaryotic organisms. So that includes the protoctists, fungi, plants, and animals. Archaea and bacteria, both classified as prokaryotes. That is because they lack membrane-bound organelles and a nucleus. Despite that shared classification, they exhibit distinct differences in the membrane lipids, ribosomal RNA, and the cell wall composition. And these variations contribute to the delineation between these 2 domains. So that's why they are split into those 2 different groups. Instead of it just being prokaryotes, it's actually bacteria and archaea to split that prokaryotic group up. The next taxon down from domain is kingdom, and we can split that into 5 different kingdoms. We have the prokaryote, the protoctistae, the fungi, plantae, and animalia. So the prokaryote, that is our unicellular, no membrane-bound organelles, small ribosomes. They have a ring of DNA with no associated proteins, it has no feeding system, and it absorbs nutrients across its surface, and some of them can actually photosynthesize as well. The protoctists are also unicellular, but they have a nucleus and membrane-bound organelles, and sometimes have chloroplasts, some have cilia, flagella for movement as well. Nutrients are absorbed either by creating it from photosynthesis or ingestion, or some of them organisms are parasites, so for example malaria. Fungi can be unicellular or multicellular, so unicellular examples are yeast, and have a nucleus, membrane-bound organelles, no chloroplasts, cannot move, they have a body made of threads or hyphae, nutrients are absorbed from dead and decaying matter, so they are saprobionts, and they can store carbohydrate food as glycogen. Plants are multicellular, have a nucleus, membrane-bound organelles, have chloroplasts, and animals, multicellular, have a nucleus, no chloroplast, no cell wall, but that links back to what you would have learnt in the cell structure topic. Viruses are acellular, so they do not fall under any of these classification systems. They don't fall within the 3 domains because they're not considered living. They're classified by the type of nucleic acid they contain, so RNA or DNA, and whether it's single-stranded or double-stranded, but they don't get classified following these classification systems because they're not living things. So next then we move on to biodiversity. And here's a selection of the key terms you need to know linked to biodiversity, which would be great to turn into flashcards to help you to remember. So populations, this is groups of organisms of the same species living in the same habitat at the same time. Habitat is part of an ecosystem in which particular organisms live. Community is all the populations of different species in the same area at the same time. Ecosystem is a community and the non-living components of the environment. A niche is an organism's role in an ecosystem, but also its habitat. So that's often two marks to know it's the organism's role and its habitat. The carrying capacity is the maximum population size an ecosystem can support. And if there are more individuals than that carrying capacity level, then individuals will start to die because there'll be a lack of resources. Abiotic factors are the non-living conditions of an ecosystem. So things like rainfall, light intensity, pH of the soil or pH of water. And biotic factors is the impact of interactions between organisms, so predator-prey relationships and competition between organisms. So the way to measure biodiversity are these three options here. You can either measure the number and range of different ecosystems and habitats. And the reason you can use that is if there is a broader range of habitats and ecosystems, that will attract a greater range of animals and a greater range of plants would be present. So that would mean the greater biodiversity. You can measure the number of species and their relative abundance, because that would indicate a greater biodiversity. And measuring genetic variation as well can be used. So essentially it's measuring habitats, number of species, or genetic variation. And if we're going to be looking at the number or type of species in an area, we'd use sampling. And it would be too time consuming to literally measure the biodiversity of an entire area, meaning counting every single individual of every species. So instead we'd do sampling, which is when you measure only some of the population, and we use this to represent the entire population. But for this to be an accurate representation, we have to make sure our sample is representative. So therefore it has to be a large sample. And if it's an area with a uniform distribution, we must randomly sample as well to avoid bias. So a bit about sampling then. We're going to go through how you could sample the slow or non-moving organisms and also the moving or motile organisms. So we'd use a quadrat if it's slow or non-motile and mark release recapture if it's motile. Uniform distribution would use random sampling. Uneven distribution would use systematic sampling, which is using a line transect. So we're going to go through random sampling, systematic sampling, and mark release recapture, what each of those methods are, starting with the sampling using a quadrat. So random sampling, the method that we would use is lie two tape measures out at right angles to create a virtually gridded area. You then use a random number generator to generate two coordinates. And if your coordinates were, let's say, 9 and 11, you would walk up 9 meters and walk across 11. And where you meet is where you'd place your quadrat. And when you place your quadrat, you can then either collect the data in forms of density, percentage cover, or frequency, which we'll come back to what that means. And because it has to be representative, we need to do a large sample. So to repeat that process at least 30 times and then calculate a mean for the area. Now, in contrast, we could do systematic sampling, and that is what we'd use if you have an unevenly distributed area. So if you were looking at a change over distance, so for example, on a sandy or rocky shore or across a path, instead of having two tape measures at right angles, you would place one tape measure going through those different areas. So you would definitely sample what is present at each position in that uneven distributed area. And that one tape measure is known as a line transect. And a belt transect is when you'd place a quadrat on the line. So it's always going to be a belt transect that you would use. But you could either do a belt or interrupted belt. And interrupted is when you place the quadrat at intervals. Belt is when you place it at every single position along your tape measure. And we'd usually use interrupted because it's more time efficient and you're not actually gaining necessarily any extra information if you're placing it every single position. You're better off spacing it out at a distance. That means you will capture where all the changes are occurring. So our method description for this would be place the tape measure at a right angle to, in this case, it'd be the shoreline because that is where we are going to be positioning it so we can see all those changes. Place the quadrat every 5 meters or set intervals along that tape measure, and then you'd collect the data. You'd then repeat by placing another 30 transects in parallel along that shoreline. So I said I'd come back to these 3 terms: density, percentage cover, and local frequency. Density is when you would place your quadrat and you'd count every individual plant. So you can do that if you don't have too many to count and it's easy to identify one individual plant. And that would give you an accurate answer because you're literally counting the number that are in that quadrat. However, some plant species, it's hard to identify an individual plant, like moss, for example, or maybe there's just too many to count accurately, like grass. And in those instances, you'd either use percentage cover or local frequency. And for these ones, you would typically use a quadrat, which has got 100 squares within it. Now, I'm not going to literally draw 100 here. I'm just going to draw some grid lines so we get the concept. But there would be 100 squares that you would use. Local frequency is when you would just simply count how many of those 100 squares have the plant of interest in. And however many out of 100, that is your local frequency as a percentage. So if you had 5 out of 100, your local frequency would be 5%. Percentage cover is when you actually have to estimate what percentage of your quadrat is covered. So this time it's not just is it present in a square or not, it's whether you think that full square is covered to be able to count it as a full percentage. So we then go on to how you would sample moving organisms, because we can't use a quadrat because they'd move away before you could count them. And we use the mark, release, recapture method. And here is the method for this. You would need to capture an initial sample of the population. You would then mark them, and we'll talk more about the ethics and the practicalities of that mark. And then you release them back after you've marked them, making a note of how many you caught and marked, and you need to make sure you release them and then leave them to randomly disperse for a long enough period of time that they do evenly disperse. You don't need to state a period of time, you just need to say it's long enough that they randomly disperse. After that period of time, you then set your traps again, collect a second sample. And this is then when we can do our calculation, because we would look at, from that second sample, what is the total number that we captured and how many had the mark on them indicating they had been recaptured. So the calculation that we use for this is the Lincoln index, and that is provided for you in the exam. You just need to know how to use this Lincoln index. So N is our population size estimate, and M is the total number of individuals marked in the initial sample. C is the total number of individuals captured in the second sample. We multiply those together and then we divide that by the number of recaptured individuals from that initial sample, which means the number that have the mark in them in that second capture. And this will then give us our population estimate. So if we think about an example of this, we've got 20 butterflies were initially caught and marked. We then had a second sample captured in which 22 were caught, 9 of which had the markings. So we would do 20, which was our initial sample, which is M, times 22, which is the number captured in the second sample, which is C, divided by 9, which is R, the number recaptured, and that comes to 48.89. Because we're estimating the number of individuals, we need to round to a whole number, so 49 butterflies would be our estimation of the population size. So going back to the concept of how we mark them, it has to be a mark that is all weather resistant and it's not going to come off, because otherwise you're going to lose the mark and you won't know that you've initially caught them. But you need to make sure that the way you capture them and mark them isn't going to cause any permanent damage or harm to the organism. So if you're going to use a paint, it has to be a non-toxic paint. You need to make sure however you tag them isn't going to increase their chances of being spotted or caught and eaten by a predator. and it mustn't interfere with their courtship ritual or chances of reproduction. Final thing linked to this is your estimate may not be accurate, and that is because of the assumptions linked to the formula. We are estimating a population size between our first sample and then when we recapture and do a second sample. But we're assuming that the population size is constant in that time. So that means we're assuming that there were no births, no deaths, and no migration. And the reality is that's probably very unlikely. There would be some change in the population size. The other reason it might be inaccurate is we said that when we release the individuals after being marked, we leave them for long enough to distribute evenly in their habitats. But the reality is animals don't evenly distribute across their habitats. They're more likely all to huddle together in favourable locations, so near a water source, near a food source, near shelter. And then in that way, you aren't necessarily going to be getting an accurate representation of all the animals present, because wherever your traps are, you might capture lots of them because that's where they happen to huddle, or none of them because the trap's in a place they don't tend to huddle. Whenever collecting data like this and analysing data, we should always use a statistic to be able to see if we have a significant difference or correlation. And when analysing a relationship between two continuous variables, so for example, how a particular abiotic or biotic factor affects the distribution or abundance of a species, like you might be investigating when you're sampling, that would be the pattern of a correlation. And we would need to know whether we have a significant correlation. So you might be looking at, is there a correlation between the light intensity and the abundance of a particular species? And that is when we'd use the correlation coefficient, Spearman's rank or Pearson's linear. Those are two statistics that you would have the formula provided for in an exam to be able to see if your patterns or correlation was significant. Now, another way to measure diversity is using the index of diversity. And this combines both the number of different species and the population size of each within a community. And it uses this formula here, where D is the diversity, capital N is the total number of organisms of all species, and lowercase n is the total number of organisms of one particular species. And this formula would be provided for you in the exam. You just need to know how to use the Simpson's index of diversity. And the key thing is knowing that you have to do the sum of lowercase n divided by capital N squared, meaning you have to work this out for every species present, and then you add that together for all of the species present. Finally, then we go on to conservation. And extinction is when all individuals of a species have died out, and this could be the result of climate change, competition between species and one species being outcompeted, hunting or overhunting by humans, degradation or loss of habitat as well. And all of those, extinction, or even if you haven't got extinction, would result in a decreased biodiversity. So climate change is the changing of the environmental conditions and habitats that results in them being uninhabitable for species, and therefore they're not able to survive there. It's also causing polar ice caps to melt and flooding, which results in the destruction of habitats, which can also lead to extinction. Habitats could also be destroyed through human actions, such as deforestation, either for the timber or for having land for agriculture or for building. Overhunting is another human impact that can cause extinction, which has been seen historically when humans have hunted flightless birds to extinction, like dodos. Interspecific competition, this is when two different species are competing for the same limited resource. One species will outcompete the other, which can result in the extinction of the species that is unable to access the resource for survival. So humans can put actions in place to try and reduce this extinction and therefore maintain biodiversity. And that's what conservation is. And it's really important that we do conserve species and biodiversity for various reasons. Number one is an ethical reason. All organisms have the right to live, and conservation helps to ensure humans are not preventing this. Conservation also helps ensure that future generations are able to experience natural ecosystems. There's a social side as well. People enjoy the outdoors and it provides many physical and mental benefits to people having a rich biodiversity. And the last one is the economic advantage. We get many medicines, food, clothes, timber, and also tourism from sources in the natural ecosystem. And that would all be lost if we did not conserve our biodiversity. So the roles of different ways then to try and conserve our biodiversity. We're going to look at the role of the zoo first, and that can be conservation breeding. Zoos play a vital role in breeding and maintaining populations of endangered species by breeding them in captivity, acting as genetic reservoirs, and that prevents extinction. Zoos also provide education and awareness to people, so they educate the public about endangered species and conservation efforts, as well as other outreach activities. They also conduct research on endangered species behaviour and reproduction to try and have better conservation strategies both in captivity and the wild. There's also species reintroduction. So some zoos participate in species reintroduction programs where captively bred individuals are then later released into the natural habitats to bolster the wild populations. We also have botanical gardens, and this helps with plant conservation. So botanic gardens preserve endangered plant species through ex situ conservation, maintaining living collections of plants for research, education, and species recovery efforts. Seed banks is when botanical gardens house seed banks, which is where seeds of endangered plant species are stored under controlled conditions for long-term preservation and future restoration projects. And then again as well, research and education is going to help educate the public about the importance of plant conservation and biodiversity. Next then we're going to have a look at the role of national parks and marine parks, which are conserved areas. So by having these conserved areas, we have habitat protection. National parks and marine parks provide protected areas for endangered species, safeguarding their habitats from human activities such as destruction, pollution, and overexploitation. We also get species conservation. These areas support populations of endangered species, allowing them to thrive and recover without human interference, whilst also preserving ecosystem integrity. Ecotourism and recreation, this is where we've got conserved areas that contribute to local economies through ecotourism and recreational activities, raising funds for conservation efforts and fostering public support for those protected areas. You can also do conservation through frozen zoos, which is where we have cryogenic preservation facilities. And this helps with genetic conservation. So frozen zoos is where you store genetic material such as sperm cells or egg cells or embryos or tissue samples from endangered species in cryogenic facilities. And that preserves their genetic diversity for future conservation initiatives. You also have genetic research, and these facilities support research on reproductive biology, genetics, and genomics of endangered species, which helps to inform breeding programs and population management strategies. And then we've got the emergency response. So cryogenic preservation facilities serve as an emergency backup in case of catastrophic events, disease outbreaks, or other threats to species survival, allowing for rapid response to recovery efforts. Now, seed banks we've actually talked about a little bit already, so this is just a brief overview going through it in a bit more detail. So you can pause, read through this and add to your notes, because it's essentially what we've just gone through with the concept of seed banks. Now, invasive species, these are non-native organisms that have been introduced to an ecosystem and have the potential to cause harm to the environment, economy, and even human health. And a key example of that is the cane toad, which is an invasive species in Australia. It was native to Hawaii, but when it was brought to Australia, it outcompeted many other amphibian species, and the toxin that they produced through their skin also killed predators such as snakes that ate them. These species, which are invasive species, often exhibit rapid population growth and spread, outcompeting the native species for resources, and they disrupt the natural ecosystem. And due to their ability to thrive in the environment and the fact that they lack natural predators, means that these invasive species can become dominant and alter the entire structure of the ecosystem. So it's really important that for conservation, we control invasive species to try and prevent the biodiversity loss that they might cause, the ecosystem damage, and the economic damage as well. Next up, we've got our IUCN and CITSs, or sites in conservation. So what these are then is the International Union for Conservation of Nature. This serves as a global authority on the status of the natural world and the measures required to safeguard it. So among its key functions, the IUCN conducts assessments of the conservation status of numerous animals. It uses classification systems and it evaluates risks of species extinction by monitoring their population. We also have the information that they provide is made accessible online through the red list of threatened species. Species are assigned to categories based on their conservation status as an indication of how at risk they are of going extinct. Then we've got the Convention of International Trade in Endangered Species of Wild Fauna and Flora, which was established in 1973, and that was initially signed by 145 countries to regulate the trade in endangered species or their products, such as furs, skin, or ivory. So since those sites started, more countries have joined this international agreement, and sites categorise species into 3 groups based on the evidence presented regarding their conservation status. Expert committees review and update these lists, although concerns have been raised about the effectiveness of this. In some cases, listing a species actually has a negative impact because if something is considered to be even more rare and at risk of going extinct, that increases its value on the black market, leading to an increase in illegal hunting and trade of those organisms and therefore threatening their survival even further. So despite these challenges, CITES plays a crucial role in regulating the international trade in endangered species and raising awareness about the importance of conservation efforts. Topic 19, the final topic, is all about gene technology. So genetic technology, and we're starting with the principles of genetic technologies. Recombinant DNA technologies is the technology of combining different organisms' DNA together to create what is known as recombinant DNA. And this is created by artificially joining together DNA of 2 different species. This is needed for scientists to manipulate and alter genes to improve industrial processes, for medical treatments as well. And then transferring DNA fragments between or within species is possible because the genetic code is universal. Transcription and translation also work the same in all organisms. So what we mean by that is the same triplet of bases code for the same amino acid in all organisms. So you can take the gene from one species, insert it into the DNA of another one, and that same protein will still be created because the genetic code is universal. And protein synthesis, transcription and translation, will work the same way. Genetic engineering is the manipulation of an organism's genetic material, introducing a new gene, and therefore causing that organism to code for a protein that results in the desired traits or characteristics. The process involves precise techniques to modify specific genes within the organism's genome. And one common method in genetic engineering involves the transfer of a gene from one organism to another, resulting in the expression of that gene in the recipient organism. This transfer of genetic material can lead to the production of new proteins or the modification of existing cellular functions, but ultimately it influences the phenotype of the organism. So this is just a summary here of some recombinant DNA technologies. Creating DNA fragments, genetic fingerprinting, and genetic counselling. And we're going to be looking at this branch first of all, how you can create DNA fragments to do genetic engineering, because all of this bit here in the blue is genetic engineering. So DNA fragments, so the first step in this recombinant DNA technology is to isolate the fragment of DNA that you want to insert into the DNA of another organism. And there are 3 methods that can be used to isolate these fragments of DNA: reverse transcription, restriction endonucleases, and the gene machine. Reverse transcription, this is when an enzyme makes DNA copies from mRNA. And this enzyme naturally occurs in viruses such as HIV, and it is reverse transcriptase. So what you'd need to do is locate a cell that naturally produces the protein of interest. That would be because that cell will have lots of mRNA for that protein. You can then remove that mRNA and combine it with the enzyme reverse transcriptase, which is the enzyme which is going to join the DNA nucleotide bases to the mRNA sequence. So we can see here we've got the mRNA molecule. You'd have complementary DNA nucleotides aligning opposite those mRNA bases, and then you'd have a single strand of DNA created. Now you'd need to have a double strand. So to make this DNA fragment double-stranded, the enzyme DNA polymerase is then used. And you'd actually then go on to use this in PCR to get multiple copies. But the key things that you need to know is that you start with an mRNA sequence. You then add in DNA nucleotides and the enzyme reverse transcriptase, and that will then create a complementary single strand of DNA to that mRNA sequence. Then you add DNA polymerase to create a double strand. Now, the advantage of this way to create your DNA fragment is the complementary DNA that is made is intron-free because it was created from mRNA, and mRNA has already had the introns spliced out. Restriction endonucleases is another method, and these are enzymes that cut up DNA. And these naturally occur within bacteria as a defense mechanism. There are many different restriction enzymes that have an active site complementary in shape to a range of different DNA-based sequences, because that is what the active site is. It's the part that is going to be complementary in shape to the substrate, which is a particular sequence of DNA bases. And that section of DNA bases is described as the recognition sequence. And in this way, restriction enzymes or endonucleases cut DNA at specific locations. Some enzymes cut at the same location in the double strand and create a blunt end, which isn't particularly useful because if you have a blunt end, it's really hard to join the DNA back together. But other restriction endonucleases cut to create staggered ends and exposed bases. And that's what we can see here with this restriction endonuclease, HIN3. It cuts and creates this staggered end, and therefore we have what's known as sticky ends, called sticky because you've got these exposed bases which would then be able to align opposite their complementary bases, and in that way it's a bit like they're sticky because they can join together more easily. So you get these cuts at palindromic sequences, meaning it's the same one way as it is backwards, the DNA base sequence, and that's how we get our sticky ends, which can then help to insert that DNA fragment into the DNA of another organism. The gene machine, this is when DNA fragments are created in a lab using a computerized machine. Scientists first have to examine the protein of interest that they want this new organism to produce, and in that way they identify the amino acid sequence of the protein, and from that they then work backwards and work out the mRNA and DNA sequence that would be required to code for that amino acid sequence. That DNA sequence is then entered into a computer and checked for biosafety and biosecurity, that the DNA being created is safe and ethical to produce. The computer then creates small sections of overlapping single strands of nucleotides that make up the gene called oligonucleotides. Oligonucleotides can then be joined to create the DNA for an entire gene. Once you've done that, PCR is then used to amplify the quantity and to make it double-stranded. So this process using a gene machine is very quick, it's accurate, and you can also design it so it's intron-free. So that's how we create our DNA fragments. Next, then we're going to have a look at how you can clone those fragments so you get lots of copies of it. So you could do this in vivo, which means inside of a living thing. So we've talked about how you create your DNA fragment of interest. Now we can look at how you insert that DNA fragment into a vector, which is an organism that will carry the DNA into the host, and the host is the organism whose DNA you are modifying. So once we've actually used those restriction endonucleases to create our DNA fragments, and we've got our sticky ends, we actually have to modify the DNA so it can be used in transcription. So we have to add a promoter region, which is added at the start of the DNA fragment. And this is a sequence of DNA which is the binding site for RNA polymerase, which will therefore enable transcription to occur. And we have to also add a terminator region, which is added at the end of the gene. And this causes RNA polymerase to detach and stop transcription. So only one gene at a time is copied into mRNA. Once we've done that, then we insert this DNA into a vector. And a vector is something that carries this DNA fragment into the host cell. Plasmids are the most common vectors which are used, and plasmids are loops of DNA that are found in some bacteria. So that circular DNA, which is separate from the main bacterial genome, only contains a few genes. So the way that we insert it then is the plasmid is cut open using the same restriction endonuclease that was used to cut out the gene of interest. That creates the same sticky ends, and therefore you're going to have sticky ends complementary to each other on the DNA fragment and on that cut open plasmid, and therefore we can join them together far more easily. Once then we have those complementary sticky ends, the DNA fragment has been cut to create that, we can then mix them together, and the enzyme ligase is used to stick together those sticky ends, sometimes known as annealing. And ligase is catalyzed in the condensation reaction to form phosphodiester bonds between the sticky ends of the plasmid and the sticky ends of the isolated DNA fragment. So now we've got our plasmid with the gene of interest inserted into it. We need to get that plasmid into the host cell, and that's what transformation is. So the vector, which is that plasmid with the recombinant DNA, next has to be inserted into the host cell, because that is the cell that's going to express that gene to create the protein. And in order to do this, we need to make the cell membrane of the host cell more permeable. And bacteria are your typical host cells. So to increase the permeability of that membrane to get the vector in, the host cells are mixed with calcium ions and heat shocked, which means a sudden increase in temperature. And that should enable the vector to enter the host cell cytoplasm. So that has now got the isolated DNA fragment into a vector and then into the host cell. But we do have to check that the host has definitely taken up a recombinant plasmid. So that's the next step in the process, identifying transformed cells, which means identifying cells that have taken up the recombinant plasmid. So not all of the host cells, which are usually bacteria, successfully take up the recombinant plasmid. And these are the three key issues that could occur. Maybe the recombinant plasmid just didn't get in through the cell membrane. Or maybe the plasmid that did get in is one that doesn't have the gene of interest in it. Because sometimes when you cut open the plasmid sticky ends just rejoin back together and you get your original plasmid again. And then sometimes the isolated DNA fragment of interest, that actually loops around and its sticky ends stick together to make a very, very tiny plasmid, and that gets inserted instead of the recombinant plasmid. So that's why we have to identify the transformed cells to make sure before we grow the bacteria en masse to create that protein of interest from the gene of interest, that you're definitely growing bacteria that contain the recombinant plasmid. So the way this is done is using marker genes. So marker genes on the plasmid is how we identify which bacteria has successfully taken up the recombinant plasmid. And the common marker genes that are used are marker genes for antibiotic resistance, or coding fluorescent proteins, or coding for enzymes. And that's because these make it easy to identify whether the bacteria has that gene in it, and therefore if it does, it means it must have the plasmid within it as well. So another concept that you need to be aware of within this is that gene editing occurs, and this is a precise and targeted form of genetic engineering that involves the modification of DNA at specific locations within an organism's genome. And unlike traditional genetic engineering methods that typically involve the insertion of entire genes, gene editing allows for the precise insertion or deletion or replacement of DNA sequences at precise locations. And this precision is achieved using specialized tools such as CRISPR-Cas9, which acts as a molecular scissors to cut the DNA at specific sites determined by guide RNA molecules. So once the DNA is cut, cellular repair mechanisms can be harnessed to introduce desired changes to the genetic code. Gene editing techniques offer unprecedented control over genetic manipulation, and that allows scientists to modify genes with high accuracy and efficiency. This capability has numerous applications in research, in agriculture, medicine, and biotechnology, including the development of disease-resistant crops, the treatment of genetic disorders, and the creation of genetically modified organisms with tailored traits. The gene editing holds immense promises for advancing our understanding of genetics and revolutionizing various fields by enabling this precise and targeted modification to the genome. So you can also do in vitro cloning, which means not inside of the organism, and that's what PCR is. So it's a way to amplify the DNA fragments that you have. And once the DNA fragments have been isolated, they need to be cloned to create large quantities. We've already gone through how you do that in vivo in the bacteria, but in vitro is PCR, the polymerase chain reaction, which is all done in an automated machine. And in this PCR machine, you need a thermocycler, which is the machine, DNA fragment that you want to make lots of copies of, the enzyme DNA polymerase, and it's a special version of that, Taq polymerase. This Taq polymerase is taken from bacteria that are found in hot springs. So they're naturally adapted to survive at incredibly high temperatures. So the DNA polymerase, Taq polymerase, has a much higher optimum temperature, and it doesn't denature at your typical 37 degrees C. It denatures at much higher temperatures, and that enables the temperature in the thermocycler to be increased to speed up the reaction without denaturing the enzymes. Primers are needed as well to attach to the DNA fragments to enable the DNA replication to start, and DNA nucleotides are needed to create your new DNA sequences. So the method then, the temperature is first increased to 95 degrees C to break the hydrogen bonds between the two DNA strands. So we now have two single strands of DNA, and they both act as a template. The temperature is then decreased to 55 degrees C, so it's cool enough that the complementary DNA primers can align opposite those complementary bases and form hydrogen bonds to hold them in place opposite those complementary base pairs. The enzyme DNA polymerase will then attach, and the free-floating DNA nucleotides can align opposite the complementary base pairs. DNA polymerase will then catalyze the formation of phosphodiester bonds between adjacent nucleotides, and that's how we get the synthesis of the new DNA strand. That process is then just repeated. That's why it's called a thermocycler, because the temperature is changed continuously in this cycle. So 95, then 55, then 72 degrees C. And it's 72 degrees C for the final stage, because that's the optimum temperature for that Taq DNA polymerase. So PCR is fully automated, it's very efficient, it's rapid, you can make billions of copies of DNA within a few hours, and it doesn't require living cells like the in vivo did, which required bacterial cells, and therefore it's quicker and it's a less complex technique. We're then going to move on to DNA fingerprinting, which is a way to look at how closely related different individuals are based on VNTRs, which are variable number tandem repeats, which are found within the introns of DNA. And the probability of two individuals having the same VNTRs is very, very low, But the more closely related you are, the more similar your VNTRs are. And that is how genetic fingerprinting works. It's the analysis of VNTR DNA fragments to determine genetic relationships and genetic variability within a population. So here are the stages we're going to go through in genetic fingerprinting: collecting your sample of DNA to analyse, how you then extract just the DNA, digest it, separate it, and then ultimately analyse your sample. So collection, the smallest sample of DNA can be collected for genetic fingerprinting, and this could be from blood or body cells or hair follicles. And if the sample of DNA is really small, then PCR is going to be used first, so you have a large quantity. Then we need to digest this DNA, and we're going to use restriction endonucleases to cut the DNA into smaller fragments. And enzymes which cut close to target VNTRs are going to be added. To separate out the VNTR, those cut up fragments, the DNA samples are loaded into these tiny wells in a block of agar gel. The gel is then placed in a buffer liquid with an electrical voltage applied. DNA is negatively charged because of the phosphate groups, because there's a negative charge in that phosphate group. So the DNA samples will move through the gel towards the positive end of the gel. And they'll move at different speeds depending on how long the DNA fragment is, so how big that particular VNTR fragment is. And that is how it separates out. Now, the agar gel creates resistance for the moving DNA, and therefore smaller pieces of DNA can move faster and further along the gel. And this is how the different DNA VNTRs are separated. An alkaline is then added to separate the DNA from being double-stranded to single-stranded. And the hybridization stage is when DNA probes, which are short single-stranded pieces of DNA, which have been created to be complementary in terms of the base sequence of the VNTRs, those are added, and the DNA probes either have a radioactive or fluorescent label, so that we can then move on to the development stage. But this part is called hybridization because the different DNA probes are mixed with those separated strands. So you've got two different pieces of DNA joining together, creating a hybrid. So the development then, the agar gel will actually shrink and crack as it dries. So we transfer the VNTRs and DNA probes using a nylon sheet, so transfer it onto that, and the nylon sheet can then be exposed to X-rays if you've used a radioactive probe, or you can expose it to UV light if it was a fluorescent probe that was used. And that is then how we get this development of your bands that we can actually see the positions of. So the position of the DNA bands are compared to identify genetic relationships, or it could be to see the presence of a disease-causing gene, or to match an unknown sample of DNA from a crime scene. And this is one that I actually created a long time ago. We can see this was the unknown that I had to identify. Here were the different samples of DNA that I had. And from this example, I could match that the unknown was the same DNA as number 3, because we can see the bands are in exactly the same position. And that is your analysis stage. So interpreting the DNA showing the results of the gel electrophoresis, this is your analysis stage again. And if you're going to do this in a paternity test, here we have the bands from the mother, here are the bands from the child, and any band in the child that wasn't from the mother's DNA must be from the father's. And that is how you can use this in paternity tests. So you'd have to match up the bands to see which bands they have in common with the different fathers to see if that particular individual could be the father. You can also use microarrays, which are used to identify the genes present in an organism's genome and determine which genes are actively expressed in the cells. And this technology allows researchers to study a large number of genes very rapidly at the same time. So following the hybridization of DNA or an mRNA sample to the microarray, which is basically a plastic dish with lots of mini dimples in it, The data are then scanned and interpreted by a computer, and the process reveals which genes are shared between species or unique to one species or absent in both. It also enables the detection of gene expression patterns in specific cell types or conditions. So, for instance, cancer cells exhibit different gene expression profiles compared to non-cancerous cells, and microarrays are used to compare gene activity between transcribed genes, converting mRNA to complementary DNA using reverse transcriptase, and amplifying that complementary DNA if necessary through PCR. You then get these fluorescently labelled complementary DNA, which hybridize with the probes on the microarray. And the fluorescent spots are going to indicate genes actively transcribed in the cell, and with the intensity of the fluorescence correlating with the gene activity level. This has been particularly useful in investigating mutations in genes associated with breast cancer, shedding light on the underlying genetic mechanisms of disease. So by providing the insight into gene expression patterns and activity levels, these microarrays offer invaluable information for understanding complex biological processes and facilitating advancements in genomics and biomedical research. Now, databases are also used, so research into the genes present in various organisms and their expression generates lots of data. And this data has to be stored digitally in a database. And that encompasses nucleotide sequences of genomes or individual genes, as well as amino acid sequences. And this field responsible for collecting, processing, and analysing all of that information is known as bioinformatics. And software developers actually play a crucial role in creating these systems for storing, searching, retrieving, and analysing this data. So computer technology facilitates the collection, analysis, and also internet-based access to this information. Notable databases include Ensemble, which houses genomic data of eukaryotic organisms like humans and zebrafish and mice. UniProt, which contains information on protein sequences and functions, and the Protein Data Bank, which provides details on protein sequences and structures. And these databases are essential resources for researchers, providing valuable insights into genetics and protein biology. There are also selection and retrieval tools, such as the Basic Local Alignment Search Tool, or BLAST for short, enabling researchers to compare sequences and identify similarities with existing database entries. Comparison between sequenced genomes, such as the human genome with that of a fruit fly or nematode worm, has revealed evolutionary relationships and common ancestry. So it's been really, really useful. Model organisms like Drosophila are instrumental in studying gene functions and developmental processes due to how easy their genetics is to work with, but also how well characterized their genomes are. And that information from their genome helps us understand the human gene function and disease mechanisms as well. So although it's not humans we're working on, we still gain valuable information to help us understand diseases within humans. So this vast genomic information stored in databases has so many practical applications. So it's been used in malaria research, for example, where gene sequences have been utilised in vaccine development as well. Additionally, databases like CFTR2 provide accessible information on genetic variants associated with diseases like cystic fibrosis, and this helps healthcare professionals in diagnosis and treatment decisions. Then we go on to 19.2, genetic technology applied to medicine. So there are advantages of using this recombinant technology. Genetic engineering enables the production of recombinant human proteins, offering several advantages in the treatment of diseases. And here are just 3 examples of proteins that have been created using bacteria, yeast, or even mammalian cell cultures. And using these bacteria, yeast, and mammalian cell cultures, producing these human proteins has several advantages. First of all, these cells are uncomplicated nutritional requirements because they're very, very basic cells. You get large quantities of the desired protein being generated. That means you've got lots of this medicine available. You can do this in facilities that require minimal space because these are single cells rather than having entire multicellular organisms. The process can be conducted almost anywhere globally, and also the protein extraction from animals or blood collection from multiple donors would therefore minimise practical and ethical concerns because you're not having to do that, you're just taking it from a single-celled organism that is producing it. So other human proteins like the factor VIII and the adenosine deaminase are produced using similar technologies. So we've got that factor VIII is crucial for blood clotting, and it's synthesized by genetically modifying hamster cells. And by inserting the human factor VIII gene into the hamster kidney and ovary cells cultured in fermenters, a constant supply of factor VIII is produced, and that eliminates the need for blood donations and therefore the associated infection risk and the availability of the blood. Adenosine deaminase is essential for T lymphocyte function and can be produced using genetically modified bacteria, E. coli, or cell cultures derived from insect larvae. This enzyme therapy has been successfully utilised for over 2 decades to treat SCID, or severe combined immunodeficiency, highlighting its long-term efficacy as an alternative treatment option. Now, genetic screening is when you would have your DNA tested to screen whether you have the gene or an allele linked to an increased likelihood of developing a disease, or it could be to see if you will definitely develop a particular disease. So we've got a whole list of advantages here of why you might want to do this. You'd get an insight into an individual's genetic makeup. It might help you to prevent the development of a disease or get better management of it. It could result in the early detection of certain diseases, such as breast cancer, where early detection is essential for increasing survival rates. You can make informed decisions about your health. Early diagnosis in general can help with personalised treatments for certain diseases, such as Huntington's disease and cystic fibrosis. It will help with preventative measures. So certain diseases you won't definitely develop if you have the associated gene or allele, but if you modify your lifestyle, you're then going to reduce the likelihood of developing it. So overall, it can help to improve patient outcomes and also therefore reduce healthcare costs. Gene therapy is a way that we could try and treat genetic diseases as well. And it involves correcting faulty genes or introducing functional ones within the genome to override the affected cells. So for example, severe combined immunodeficiency and inherited eye diseases have been targeted for gene therapy interventions. So in SCID, where the deficiency is linked to ADA, which is comprising the immune function, gene therapy involves restoring normal ADA activity, and that will then give long-term benefits. In the inherited eye diseases like LCA, gene therapy restores vision by introducing functional alleles of the RPE65 gene into the affected retinal cells. And these examples highlight the potential of gene therapy in providing long-lasting solutions for genetic diseases, therefore improving patients' quality of life. Now, so far we've only highlighted all the positives with these technologies, but there are social and ethical considerations linked to this as well. So while genetic screening does empower individuals to make these informed decisions about their health, it does link to some concerns around privacy, discrimination, and also psychological impact. Because in terms of privacy, once you have screened yourself, there's the question of, well, who then gets access to that information? And it could be, for example, maybe insurance providers might get access, and therefore they wouldn't give you health insurance anymore because they know that you are at higher risk of developing a particular disease. You might get discriminated from an employer if they knew that you are likely to develop a disease later in life. And also the psychological impact of knowing you have an allele that makes you at risk of developing a disease. That can play heavily on someone's mind. So gene therapy does pose challenges related to safety, efficacy, and also equitable access to treatment as well. The ethical dilemmas arise regarding the manipulation of human genetic material and therefore the potential for eugenics and the allocation of resources. So we do have to strike a balance between the benefits of these scientific progresses, whilst also considering the ethics and the social side of the considerations as well. And lastly then, it's genetically modified organisms in agriculture. So genetic engineering is one technology that could really help with the global demand for food, because we could enhance the quality and productivity of farmed animals and crop plants. So for example, GM salmon have been developed to grow faster and more efficiently, offering a potential solution to meet the increasing demand for seafood. Also, we've got herbicide-resistant soybean varieties have been engineered to tolerate herbicides, which enables farmers to control weeds more effectively and therefore increase their crop yield. We've also got insect-resistant cotton varieties created by incorporating genes into Bt bacteria, which produce toxins lethal to certain pests, and therefore it would kill the pests without needing to apply chemical pesticides, and therefore it's a more sustainable agriculture. So this shows how you could use genetic engineering to improve food security, sustainability, and also economics. But again, there are downsides to consider linked to the ethical and social implications. So the use of genetically modified organisms to produce food in this way has one issue around GM cultivation. So unintended effects maybe of non-targeted organisms. So maybe it does actually affect other organisms, not just the crop plant of interest. It could result in maybe biodiversity loss if it causes the death of other plants. And also you might get herbicide-resistant weeds and pest populations. There's also concerns that this is a relatively new technology, so we don't know the long-term effects of humans eating GM food. And there may well be no long-term effects, but we actually don't know because we haven't tested it. We've got social economic factors as well, because these seeds are very expensive. So therefore, they become too expensive for poorer farmers or poorer countries to be able to afford to buy these seeds to get these GM crops. And then we've got ethical debates as well, which is looking at the moral implications of manipulating living organisms and the potential consequences it might have on animal welfare, ecosystem integrity, and future generations as well. So that takes us to the end of the genetic technology topic. I hope you found it helpful. If you did, don't forget to subscribe.